Study about xylem vessels. So what is meant by xylem vessel? So here the structure which you are seeing here, this is called as xylem vessel. This one you can call it as xylem vessel. So it is a long cylindrical tube-like structure made up of many cells which are called as vessel members, each with lignified walls. What is meant by lignified walls, ma? It means walls they are made up of lignin. Okay, so they are made up of lignified walls, and protoplasm is absent in this condition. Okay, so what is meant by protoplasm is absent? It means they are also dead cells. So protoplasm is absent in this condition. So which always represents that the cells are dead cells. So vessel members are interconnected through perforations in their common walls. Okay, so in the figure, so this is the structure of the vessel. This is how vessel look like, and you can see small holes they are present here. Okay, so very small holes are present. So these small holes they are called as perforations. Okay, and through these small holes only these the cells of vessels they are connected to each other. So vessels are a characteristic feature of gymnosperms. It means in the case of angiosperms. In the case of angiosperms only you can find these vessels, and in the case of gymnosperms you cannot find the vessels. So vessels are a characteristic feature of only angiosperms only. In gymnosperms you don't find any vessels. Clear? So then we study about the next one which is called as xylem fibers. Okay, so these xylem fibers they have highly thickened walls. So xylem fibers, where you find this xylem fibers? So this is the structure of xylem fiber. So here you can find the xylem fibers. So these are the xylem fibers. Clear? So walls are very thick, highly thickened walls are present, and obliterated central lumens are present. So what is meant by obliterated central lumens? Well, let us see here. So walls are highly thickened and lumens get compressed. So that is the meaning of obliterated central lumens. Clear, ma? So highly thickened walls are present. So as a result, the lumens means the gaps, the transport system, the gap which is present inside the xylem fibers, they are highly compressed. Clear, Nama? So that is what is called as obliterated central lumens. Clear? And the next property is that they may be septate also and they may be aseptate also. Septate means separations may be present and separations may be absent also. So what is meant by separation? So previously already we have studied, Nama. So for example, if this is a structure, clear, ma? If the separations like this, then they are called as septate. And inside these separations you can find a nucleus also. And aseptate means, so just a structure is present like this. So nuclei they are arranged at different positions without any separations. They are called as aseptate. So xylem fibers may be septate also, they may be aseptate also. And because highly thickened walls are present, their lumens are highly compressed. This is called as obliterated central lumens. Clear? Okay. Then we study about xylem parenchyma. Okay, ma? So this layer, this last layer which you are seeing here, this is called as xylem parenchyma. This one is called xylem parenchyma. Clear? So therefore, let us see what is the function and what is the construction of this xylem parenchyma. Ma? So living cells. So xylem parenchymatous cells, they are first of all they are living cells, and thin walled cells with cellulosic cell walls. Okay, ma? So their walls are very thin and they are made up of cellulose. So they store food material like starch or fat and other substances like tannins also. What is the function of xylem parenchyma? They have the ability to store the food material. So radial conduction of water takes place by ray parenchymatous cells. Clear, ma? So through which cells conduction of water takes place? Conduction of water takes place through ray parenchymatous cells. So what is meant by ray parenchymatous cells? These are the radial cells. For example, if this is the wooden structure which you are seeing, cross section of the stem, then these ray parenchymatous cells they will be traveling in radial fashion. So this type of cells you can call it as ray parenchymatous cells, and because they are arranged in radial fashion, conduction of water takes place radial. They are called as xylem parenchyma. Clear, Nama? Okay. Then uh, next we said primary xylem. So primary xylem is what? The xylem which is formed at the initial conditions of the plant. It is called as primary xylem. So when cambium is present between xylem and phloem, then you can find a secondary xylem also. Clear? No? That we will study later. So first of all, this primary xylem, we are dividing this one into two parts or two types. What are they? First one is called the protoxylem and the second one is called as metaxylem. Okay, ma? Protoxylem means what? The first formed primary xylem, and the metaxylem is called as laterally formed, that is a later formed primary xylem is called as metaxylem. Okay, ma? So proto means initially, meta means in the latter conditions. So primary xylem you can divide into two types, protoxylem and metaxylem. Clear, no? Next, xylem. Clear? So here in stems, so protoxylem lies towards the center and metaxylem lies towards the periphery. This is called as endarch. So what is meant by endarch? Endarch is a condition in which, so this protoxylem, it lies towards the center. And what is meant by pith here? So this region we know it is called as pithnoma, the centermost region, the undifferentiated region, that is called as pith. Okay, the soft region which is responsible for storing food material also, that is called as pith. Okay, so this pith, so towards this pith if a protoxylem lies, and towards the periphery, periphery means what? Outer structure. This structure is called as periphery. Okay, and towards the out structure if metaxylem is lying, then this type of arrangement is called as endarch. Okay, ma? That arrangement we can call it as endarch, and this you can find in the case of stems. In stems you will find the endarch. Then we are studying about exarch. What is exarch? The exactly opposite condition. So towards the pith you can find metaxylem, and towards the periphery you can find the protoxylem, that is called as exarch. And where you find this exarch? Exarch you will find in the case of roots. Okay, that is the condition. Okay, ma? Then we are studying about phloem bast. Okay, ma? So let us see what is meant by phloem bast. Okay, so phloem, or you can call it as bast. So it transports food materials from leaves to other parts. Okay, ma? So basically, vascular system we can divide it into two types. No, one is xylem and the second one is called the phloem. So what xylem does? Xylem transports water and other nutrients to the plant through the roots. Clear, no? And what phloem does? Phloem, it supplies the food material which is produced in the leaves in the form of starch through photosynthesis, and it is supplied to different parts of the plant. So that arrangement we can call it as phloem bast, or you can call it as phloem, or you can call it as blast also. Clear, no? Okay. So in angiosperms, phloem is composed of, so let us see. So in angiosperms, phloem is composed of what? It is composed of sieve tube elements. So it is composed of companion cells, Phloem Parenchyma and Phloem Fibers. So these are the structures which are present in the case of angiosperms. Clear? Next, in the case of gymnosperms, you will have albuminous cells and sieve cells. So these lack sieve tubes and companion cells. So sieve tubes and companion cells, they are absent in the case of gymnosperms. So this is one difference which you should remember. Okay. So sieve tube elements are present, companion cells are present, phloem parenchyma are present, and phloem fibers are also present in the case of angiosperms. And in the case of gymnosperms, albuminous cells and sieve cells are present, but the sieve tubes and companion cells, they are absent. Now let us study each of them in detail. First one we will study about sieve tube elements. Okay, ma? So this total structure which you are seeing here, so this structure you can call it as sieve tube element. So this is called as a sieve tube element. Okay, ma? So they are long tube-like structures. Clear, no, ma? So which are arranged longitudinally. So what is their arrangement? Arrangement is longitudinal, and they are associated with companion cells. It means beside this sieve tube elements you can find one more structure. So this structure is called as companion cell, and in the companion cell you will find a nucleus also, and this nucleus regulates the functioning of these sieve tube elements. Okay, ma? So able to understand what I am saying? The nucleus which is present in the companion cells, that controls, that maintains the pressure required for the sieve tube elements. Okay, ma? So the end walls are perforated to form the sieve plates. Okay, ma? So what is meant by sieve plates here? So this plate which you are seeing, this is called a sieve plate, And in this sieve plates, you will find some holes which are present or the perforations which are present. So mature sieve element have a peripheral cytoplasm and a large vacuole and lacks the nucleus. Clear, Nama? So mature, that is fully developed sieve tube elements, it has a peripheral cytoplasm. It means cytoplasm you can see only on the outer walls of the towards the inner walls of this sieve tube element only. At the middle you don't find any cytoplasm. So a large vacuole is present and it lacks the nucleus. What is meant by vacuole? So vacuole is a membranous covering which is present inside the cell. So it maintains the pressure difference also. Clear, ma? So it is a sphere with fluid and molecules inside a cell. So only in the case of plants only you can find a vacuole. Clear, ma? It stores water and maintains pressure in a plant. So for example, if you take a plant cell where vacuole is present, so this vacuole covers nearly 70 to 80% of the volume of the cell. So are the major components like water, so different cells and molecules, they are present inside this vacuole inside the cell. So for example, if this is a cell, so in the middle you can find this vacuole. Okay, ma? It covers nearly 70 to 80% of volume of the cell. So therefore, in this sieve tube elements also you can find a vacuole and it lacks the nucleus. Are you able to understand? And outside this vacuole you can find the cytoplasm. So that is why it is called as a peripheral cytoplasm. Okay. So the functions of sieve tube elements, clear, Noma? They are controlled by the nucleus of companion cells. So already we have discussed about this one. So the functions of the sieve tube elements is controlled by the nucleus which is present in the companion cells. Clear? Okay. So okay, ma? Next see. So the first formed primary phloem consists of narrow sieve tubes. Okay, ma? So the first formed primary phloem, which is called as phloem, it consists of narrow sieve tubes. So narrow sieve tubes you will find only in protoplasm. So the latter formed phloem, which is called as metaphloem, has bigger sieve tubes. Okay, it means narrow sieve tubes, they are present in the initial stages of the what you call phloem, which is called protoploem. And in the metaphloem, that is the secondary formed phloem, the latter formed phloem, there you will find bigger sieve tubes. Okay, and what is the function of phloem? Already we have studied now. So conduction of food materials from the leaves to other parts of the plant, that is the function of phloem. Okay, so next to see. So next one which we are studying is companion cells. So this structure which you are seeing here, okay, this structure, this is called as companion cell. So specialized parenchymatous cells associated with sieve tube elements. So therefore, what is the nature of cells present in this companion cells? The simple cells which are called as parenchymatous cells. So they are associated with sieve tube elements. So sieve tube elements and companion cells are connected by pith fields present between their common longitudinal walls. So what is meant by pith fields here? Okay, ma? So here in this region, you are seeing a small pit-like structure, no? Okay, ma? So you are seeing small pit-like structures here. They are called as what you call pit fields. Clear, nama? So companion cells are connected by pit fields present between their common longitudinal walls. Okay, so sieve tube elements and companion cells, they are connected through some perforations, some holes, which is called as pit fields. And what is the function of these companion cells? They maintain pressure gradient in sieve tubes. So maintenance of pressure in sieve tubes is the function of companion cells. Clear, Nama? Next, phloem parenchyma. So this region total, this cell, it is called as phloem parenchymatous cell. Clear, Nama? So it is made up of elongated tapering cylindrical cells, so which have dense cytoplasm and nucleus. Okay, so what is the nature of this parenchymatous cells, ma? So in the figure you can see this is called as phloem parenchyma. So this one, this cells, so this cell is called as a phloem parenchyma. Clear, ma? This cell is also called as a phloem parenchyma cell. Okay, so it is made up of elongated tapering cylindrical cells. First of all, they are elongated cells. Okay, ma? And they are tapering cylindrical cells. Tapering means what? At the ends they become very, very narrow. That is called as ends or narrow. Okay, ma? So that is called as tapering cylindrical cells, and they are in the shape of cylinder. So which have dense cytoplasm and a nucleus. So dense cytoplasm is also present, nucleus is also present inside these cells. So cell wall is made up of cellulose and has pits through which plasmodesmatal connections exist between the cells. What is meant by plasmodesmatal connections, ma? In between one parenchyma cell and other parenchyma cell, there will be some connections through which the cytoplasm will be connected. So such type of connections is called as plasmodesmatal connections. Are you able to understand, ma? So connections through which what is connected, cytoplasm of two cells is connected. That is called as plasmodesmatal connections. Okay, ma? So phloem parenchyma is absent in most of the monocots. It means there are some monocots which have this parenchyma also, phloem parenchyma also, but in most of the monocots it is absent. Okay, but this is one point which you should understand. Now, what is the function of this phloem parenchyma? It stores food material and other substances like resins, latex, and mucilage. Clear, ma? So the storing of resins, latex, and mucilage. So this is the function of this phloem parenchymatous cells. Okay. Next, we study about phloem bast fibers. Okay, ma? Phloem fibers, or you can call them as bast fibers. Okay. So they are made up of what? So we studied that phloem is made up of four parts, no, ma? Four types, and this is the fourth one. So basically it is made up of sclerenchymatous cells. So generally absent in primary phloem. So you can find the phloem fibers or bast fibers only in secondary phloem. So they are elongated, unbranched, and have pointed needle-like apices. Apices means what? Ends. Tapering ends we have studied, Nama. So like this they have pointed needle-like apices. So cell wall is quite thick. Thickness of the cell wall is large. At maturity, these fibers lose protoplasm and become dead. So it means initially they were living only, but when they mature, they become dead and they lose the total nucleuses as well as the cytoplasm. So they become dead. So phloem fibers of jute, flax, and hemp are used commercially. So for commercial purposes, you will use the phloem fibers of jute, flax, and hemp. So mechanical support and protection of soft tissues, that is the function of phloem fibers. So the soft tissues of the plant are protected by this phloem fibers. Okay, and this is the overview of what we have studied up to here. So plant tissues, you can divide them into meristematic and permanent. Okay, ma? Meristematic, again you can divide into apical meristem, intercalary meristem, secondary meristem. Clear, ma? Permanent tissues you can call them as simple and complex. Apical meristem you can divide into root apical and shoot apical. Simple permanent tissues you can call them as parenchyma, collenchyma, and sclerenchyma. And complex you can call it as xylem as well as phloem. Okay. So this is what we have studied up to here. So in the next class we will study next tissue system we are studying. Clear, Nama? So based on structure and location, tissue system can be of three types. What are the three types of tissue system? Based on what we are dividing this one? Based on what we are dividing? Based on structure and location, where they are located. Clear, Nama? So first one is epidermal tissue system. Second one is ground tissue system, or you can call it as a fundamental tissue system. And the third one is called as vascular tissue system or conducting tissue system. Are you able to understand, ma? Epidermal tissue system means what? Which is present on the outer covering of the plant. So there will be an outer covering to the plant, no, ma? So that total outer covering we can call it as epidermal tissue system. Okay, ma? And there are some tissues and cells which are used for transportation of water and food materials. They are called as vascular. Are you able to understand? So, excluding these tissue system, epidermal system and vascular system, remaining all the tissue system, they are called as ground tissue system. Are you able to understand, ma? So, therefore, how can you define a ground tissue system? Ground tissue system means all tissues except epidermal and vascular. Okay, ma? Remaining all the tissue system, you can call it as a grounded tissue system or fundamental tissue system. Are you able to understand? So here in the figure also he tried to give some explanation. See here. So for example, if you see the leaf, if you cut the leaf into two parts, then here we are cutting and you are seeing a green color layer, no? Above that one you are seeing a green color layer. What is that green color layer? What is that is called as epidermal tissue system. Green color layer is nothing but epidermal tissue system. Then you are seeing black color dots and a big dot, no? So if you take this figure here, you are seeing a black color dots, no ma, in this figure. Are you able to see or not? Clear? I am discussing about these dots. Clear, no? Black color dots you are able to see, no? So what are those black color dots? They are vascular tissue system. And the remaining yellow region which you are seeing, no? In the figure you are seeing the yellow region, no? So what is that yellow region? That becomes the ground tissue system. Okay, ma? So totally tissue system I can divide into three types. Okay, one is epidermal, second one is ground, and the third one is vascular tissue system. Okay, that's it. Similarly here also you can see, for example in the stem also you can see, no ma? So this green color region that becomes the epidermal, and this black color region that becomes the vascular, and the remaining yellow color region you can call it as the uh, ground tissue system. Are you able to understand? That's it. Okay. So first of all we are studying about epidermal tissue system. Okay. So it forms the outermost covering of the whole plant body. So it comprises epidermal cells, stomata, and epidermal appendages. And what are the examples of epidermal appendages, ma? Trichomes and hairs. So trichomes and hairs, they can be used as synonyms. Both are almost one and the same. Are you able to understand? Trichomes and hairs. So on the stems of the roots, stems of the plants, you can see some hairs-like structures, no ma? So them also you can call them as trichomes. Okay. So the outermost covering is totally called as epidermal tissue system. So what are the types of cells they have? So they have epidermal cells are present, stomata are present for respiration purpose, and epidermal appendages. Appendages means extra growths in the form of trichomes and hairs are present on the epidermal tissue system. Are you able to understand? So here you are seeing this trichome, no ma? This is a head-like structure, no? This is called as a trichome. Okay. And here you are seeing this cuticle. What is meant by cuticle? Cuticle means a wax coating. A wax coating on the leaves and the stems is called as cuticle. Are you able to understand? This cuticle you can find only on leaves and stems only. You cannot find the cuticle on the roots. Cuticle is absent on roots. Are you able to understand? So this is dermal tissue cells, and these are the god cells. Clear? And sunlight is entering from here. Okay. So therefore epidermal tissue system is composed of epidermal cells. Stomata is present for respiration, and trichomes and hairs are present. Okay. So it is the outermost layer of the primary plant body. Okay, it is the outermost layer. So and it is single layered also. How many layers are present? Only one layer is present. So it is made up of elongated, compactly arranged parenchymatous cells with a small amount of cytoplasm lining the cell wall, and a large vacuole is present. So these are the properties of the cells which are present in epidermis. So what are the types of cells they have, ma? Elongated cells are present. Compactly means they are very closely packed. They are called as compactly arranged. And what kind of cells they have? Parenchyma cells. Clear, number? Cytoplasm is very less. Small amount of cytoplasm is present, and a big vacuole is present. What is meant by vacuole we studied in the last class, no? Okay, ma? For example, if you take this is a cell, so the water component of this total cell is enclosed by vacuole. So it covers nearly 70 to 80% of the total volume of the cell. That is called as vacuole. Clear, Nama? So outside of the epidermis is often covered with a waxy thick layer which is called as cuticle. And what is the work of cuticle, ma? It prevents the loss of water. So cuticle is absent in roots. You cannot find the cuticle in roots. This is about epidermis. Okay. So then we study about stomata. What is meant by stomata? So they are present in the epidermis of the leaves. So they regulate transpiration and gaseous exchange. What is meant by transpiration, ma? Transpiration means what? Sima, plants, they are responsible for evaporation of water also, no? If there are a huge number of plants in a particular area, then more amount of rainfall is possible in that area. Do you know that? Why? Why? Because from the leaves of these plants more and more water will be evaporated, so they may form into clouds, and when air touches the clouds, they again rainfall may happen. Are you able to understand? So this transpiration, that is the evaporation of water and exchange of gases, it is regulated by whom? It is regulated by the stomata. Are you able to understand, ma? So they are very small holes which are present at the bottom of the leaves. So if you observe the leaf under compound microscope, you can observe this stomata also. Are you able to understand? So stoma. Stoma is what? It is a plural form. Stoma is made up of two bean-shaped cells, which are also called as gourd cells. Clear, ma? But in grasses, the gourd cells are dumbbell shaped. So generally, these stoma, they are made up of what kind of cells, ma? Bean-shaped cells. It means cells will be of this kind of shape. They are called as bean-shaped cells. Cells. Clear, no, ma? But in grass, what is the shape of these stoma cells? They are bean-shaped. So here you are seeing this bean-shaped cells, no, ma? Sorry, dumbbell. In grasses, you will find dumbbell shape, and in other plants, you can find them in bean shaped. Clear, no? That's it. So let's see once what are the different parts which are present in the layer of this leaf, ma. Clear, no? So this is called stomata. This one you can call it as the stomata. Clear, no, ma? And these are the gourd cells which are present in the shape of bean shapes, and this is called as stoma. Clear, no? So here we have vascular bundles. What is meant by vascular bundles, ma? So these are the bundles which are responsible for transportation of water, transportation of food materials. So vascular bundles are present. Clear, no? Chloroplasts are present. So what is meant by chloroplast? Responsible for performing photosynthesis. Clear, no? Trichomes are present. What is meant by trichome? Hair-like structures already we have studied, Nama, which are present on the epidermis. So they are the trichomes. And here you can find the xylem layers. And this is the sheet of bundle sheath which is present here. Clear, no? And this is the cuticle. Cuticle means what? Just a very thin layer of wax is called as cuticle. So this is the upper epidermis, palisade mesophyll cells, spongy mesophyll cells, lower epidermis, cuticle, air space, and phloem. Okay, ma? So these are the different parts which are present in a leaf. Okay, and where you find this stomata, ma? This stomata you will find at the bottom of the leaves mostly. Are you able to understand? That's it. So the outer walls of gourd cells. So gourd cells, they can be of two shapes. No? It can be bean shaped also. They can be dumbbell shaped also. So outer walls of gourd cells. So that means away from the pore, away from the stomatal pore, are thin, and inner walls are thick. So this is one point which can be asked in the examination. Are you able to understand, ma? So these bean-shaped cells on the outside, that is on the structure which is away from the pores, they are very thin. But when they start coming toward the center, they become very, very thick. Okay, ma? It means that on this side, they are very thin. And on this side, they are very thick. Are you able to understand? So that is one point which you should understand. Clear, no, ma? So the gourd cells possess chloroplast and regulate the opening and closing of stomata. So the opening and closing of stomata, hmm? That's good. So the opening and closing of stomata, it is regulated by whom? It is regulated by the chloroplast which is present in the gourd cells. Are you able to understand, ma? So in this bean-shaped or dumbbell-shaped gourd cells, you will have chloroplast. And chloroplast is responsible for controlling of this opening and closing of stomata. Clear, no? Sometimes some epidermal cells near the gourd cells become specialized in shape and size. They are called as subsidiary cells. Okay, ma? So here you can find this subsidiary cells, no, ma? These cells, they are called as subsidiary cells. So here you are finding some shapeless structures here, no, ma? So they are called as subsidiary cells. Are you able to understand, ma? So what is the function of these subsidiary cells, ma? just for accommodating this stomata. For making this stomata to be present in that region, these subsidiary cells are responsible. So they are modified such that stomata is present in that particular region. Okay, ma? So such modified cells, what you call them? You can call them as subsidiary cells. Clear? So stomatal aperture, God cells and the surrounding subsidiary cells are together called stomatal apparatus. Okay, ma? Stomatal aperture means the hole which is present. So when this bean shaped cells, when they do like this, then hole will be closed. Clear, ma? And when they start contracting, then the hole comes into action. That is, you can see the hole from where gaseous exchange take place. Are you able to understand? So this stomatal aperture and the God cells which are responsible for creating this stomal aperture and the subsidiary cells, that is the modified cells. Together, what you call together, you can call them as stomatal aperture. So this is another point. So see here, one more diagram is given here. So these are the epidermal cells, and these modified cells you can call them as subsidiary cells. Then you can find here dumbbell shaped God cells. Clear, no? That's it. So, so here in the figure you can see very clearly, ma. So this is the condition when stomata is open, ma. Clear, no? This is the condition when stomata is open, and this is the condition when stomata is closed. Okay, ma. So what is meant by stoma? Stoma is a singular form. Stomata, plural form. That's it. Are you able to understand? Off करो बोल जरा. So ఈ stomata opening కి closing కి help అయ్యే cells ఏవి? These are the God cells. ఈ bean shaped dumbbell shaped God cells ఉన్నాయి కదా. So they are responsible for stomata. Clear ma? So తర్వాత మనము we have also studied about the subsidiary cells. Subsidiary cells గురించి కూడా చదివినాం కదా. What is meant by subsidiary cells? సో ఈ స్టొమాటా చుట్టుపక్కల ఉన్న కొన్ని సెల్స్ మాడిఫై అవుతాయి. వాటి షేపు సైజు మాడిఫై అయ్యి ఇవి స్టొమాటాని అకామిడేట్ చేయడానికి హెల్ప్ అవుతాయి. అవే కదా సబ్సిడరీ సెల్స్ అంటే మనం అనుకున్నది. ఇక్కడ ఉంది చూడండి ఫిగర్. ఓకేమా? సో ఇక్కడ సబ్సిడరీ సెల్స్ ఏవి? దీస్ ఆర్ ద సబ్సిడరీ సెల్స్. ఓకే, సో ఇవి సబ్సిడరీ సెల్స్ అవుతాయి. క్లియర్ మా? సో దీస్ సబ్సిడరీ సెల్స్ దే ఆర్ మాడిఫైడ్ టు అకామిడేట్ స్టొమాటా. స్టొమాటాని ప్లేస్ చేయడానికి అవి యూజ్ అవుతాయి. తర్వాత ఇంకేం చదివినాం మనము? సో స్టొమాటల్ అపర్చర్ అంటే అక్కడ క్రియేట్ అయిన ఓపెనింగ్, గాడ్ సెల్స్ అండ్ ద సరౌండింగ్ సబ్సిడరీ సెల్స్, ఈ మూడు కలిపితే దాన్ని ఏమంటాం మనము? వి కాల్ ఇట్ యాస్ స్టొమాటల్ అపర్చర్. అదే కదా మనం స్టడీ చేసింది. క్లియర్ ను? సో స్టొమాటల్ అపర్చరు, గాడ్ సెల్స్, అలాగే సబ్సిడరీ సెల్స్ ఇవన్నీ కలిపితే మనము స్టొమాటల్ అపర్చర్ అవుతుందని చదువుకున్నాం. క్లియర్? దట్స్ ఇట్. సో then we study about epidermal appendages. Okay ma? Appendages ante what is meant by appendages ma? So the extra growths which are happening in epidermis, that is called as epidermal appendages. అర్థమవుతున్నాయి కదా? So what are the epidermal appendages here? Root hairs and trichomes. So these are the epidermal appendages. Are you able to understand ma? So root hairs, trichomes, so this will be the epidermal appendages. Okay, so what is meant by root hairs here? So unicellular elongations of the epidermal cells. They help to absorb water and minerals from the soil. So root ki hairs ఉంటాయి కదా, అలాగే on the stem of the plant you can see some outgrowths, no ma? So they are called as trichomes. So generally when these appendages, when they are present on the roots, then they are called as root hairs, and same appendages when they are present on the stems, they are called as trichomes, outgrowths anamata, extra growths which are happening on the stem and the roots, they are called as appendages. Clear? So here you are able to see this trichomes, no ma? Ikkada trichomes kanipistunnai kada, so they are also appendages. Clear? So trichomes which are present on the stem and the hairs which are present on the roots, they are called as epidermal appendages. Clear? So what is the purpose of root hairs ma? What is the purpose of root hairs here? They help to absorb water and minerals from the soil. That is the purpose purpose of root hairs. And what about the trichomes? Epidermal hairs which are present on the stem, they are called as trichomes. So they are usually multicellular, clear ma? They are branched or unbranched also. So soft also or stiff also. They may be secretory also. They prevent water loss due to transpiration. Okay ma? So what are the type of trichomes which he is discussing? So they may be unicellular also, they may be multicellular also, but usually they are multicellular. Clear no? So they may be branched also or they may be single also, and they may be soft also, they may be stiff also. So they may be secretory. Secretory means what? Some trichomes, they will be continuously emitting some hormones, no ma? Some plant regulators or some gums. Some you should have seen some trees secreting some gum type of material, no? So these secretions can be done by these trichomes. So they can be secretory also, and they can prevent water loss also because of transpiration. What is meant by transpiration? Evaporation of water from the plants is called as transpiration. Clear, Nama? So the transpiration can also be prevented by trichomes. So this is the function of epidermal appendages in the case of plants. Clear? So they are studying about ground tissue system. Okay, ma? So first of all, we have studied about epidermal tissue system, Nama. Second one is the ground tissue system. And what is the third one? Third one is the vascular tissue system. Clear, no? So second one is ground tissue system. So all tissues except epidermis and vascular bundles, they are called as ground tissue system. Are you able to understand, ma? Epidermal layers, what is meant by epidermal layers? Outer layers, and vascular bundles means the bundles which are responsible for transportation of water and other minerals. Clear, Nama? So except epidermis and vascular bundles, remaining all the tissues, they come under ground tissues. అర్థమవుతున్నాయి కదా? ఎపిడర్మల్ టిష్యూస్, వాస్కులర్ బండిల్స్ తప్పితే మిగతా అన్ని టిష్యూస్ కూడాను దే కమ్ అండర్ గ్రౌండ్ టిష్యూస్. సో దే కన్సిస్ట్ ఆఫ్ సింపుల్ టిష్యూస్. వాట్ ఆర్ ద సింపుల్ టిష్యూస్? So parenchyma, collenchyma, and sclerenchyma. Already we have studied, Nama. So this ground tissues, they consist of simple tissues, parenchyma, collenchyma, as well as sclerenchyma. Clear? So parenchyma cells are present in. This is very important bit, Nama. So where is the presence of parenchymatic cells that he is mentioning here? So parenchymatic cells are present in cortex. Clear, Nama? They are present in pericycle. They are present in pith, and they are present in medullary rays in the primary stems and roots. So these are the regions where you can find parenchymatic cells. So question can be asked from this position. So they are present in cortex, they are present in pericycle. So what is meant by pericycle? It is a region which lies below the endodermis or which lies inside the endodermis. What is meant by endodermis? The innermost layer of the plant stem is called as endodermis, and inside this endodermis you can find a layer which is called as pericycle. అర్థమవుతుంది కదా? What is meant by epidermis? Outermost layer is called as epidermis. Endodermis means innermost layer is called as endodermis. And where you find this pericycle? Pericycle you will find again inside the endodermis, and inside that pericycle again you will find a soft tissue type of substance which is called as pith. అర్థమవుతుంది కదా? సో ఫస్ట్ ఆఫ్ ఆల్ విచ్ లేయర్ ఈస్ ప్రెసెంట్? ఫస్ట్ ఆఫ్ ఆల్ endodermis is present. Inside endodermis, pericycle. Inside pericycle you can find pith. So this is the construction of the plant stem. Clear, Nama? So parenchymatic cells, they are present in cortex, they are present in pericycle, they are present in pith, and they are present in medullary rays in the primary stems and the roots. Okay, ma? So what is meant by medullary rays, ma? Medullary rays means already we have studied, no? For example, if this is the shape of the stem, then these rays, they will be traveling like this. Okay, ma? From the center of the stem, these rays, they will be traveling radially. They are called as medullary rays. So what is the purpose of these medullary rays? They are again responsible for transportation of water, radial transportation of water. Are you able to understand, ma? Radial transportation means what? They are arranged on the radius only, no? So through these radial structures only, water is transported throughout the stem. So these rays, so these continuous lines, they are called as medullary rays. And these medullary rays also, they are made up of what? They are made up of parenchymatous cells. Clear? That's it. So therefore, here is there anything? Already we have studied all of them. Okay. So then in leaves, the ground tissue consists of thin walled chloroplast containing cells and is called as mesophyll. Okay, ma? In leaves also you will find ground tissues, ma. Already we have studied the cross section previously in the previous slide, no, ma? If you cut a leaf into at the middle, you can find a green color layer which is called as epidermis. You are seeing black dots which is called as vascular system, and there was an yellow region, no? That yellow region is called as ground tissue. Clear? So in leaves, ground tissue consists of what, ma? It consists of thin walled chloroplast. So chloroplasts are very thin, their walls are very thin. So, and they are called as what, ma? They are called as mesophills. So this may be another important bit which you should remember. So what is meant by mesophyll, ma? Mesophyll means what? Mesophyll means it is a ground tissue, basically. Ground tissue. And this mesophyll consists of what? This mesophyll consists of chloroplast. And what is the nature of chloroplast, ma? Their walls are very thin. So such type of cells we can call them as mesophills. Clear? That's it. And what is meant by pith here? Here I have written. Pith means what? Pith or medulla is a soft spongy parenchymatous cells which can sometimes store starch. Okay, ma? So that is the definition of pith. And what is the arrangement where this pith is present? Pith is present on the innermost layers of the stem. So it is undifferentiated region, ma. It means pith you cannot identify very easily. Until you observe very carefully only, you cannot observe this pith. So at the center of the stem center lo unde region emantam manamu, this region is called as pith. It is very soft, it is spongy, made up of parenchymatous cells, and it is sometimes it will store energy also, it will store starch also. Clear? So this is the functioning of the pith. Okay. Then we are studying about the third type of tissue system which is called as vascular tissue system. So we have studied epidermal tissue system, we have studied ground tissue system, and now we are studying vascular tissue system. Clear, Nama? Now let us study what is the importance of this one. So it consists of complex tissues like xylem and phloem. Okay, so vascular tissue system means what? Which is responsible for transportation of water and other food materials, minerals, all this called as vascular tissue system, Nama. So vascular tissue system basically made up of what? Basically made up of xylem as well as phloem. Xylem is responsible for what? Xylem is responsible for what? For transporting water. Phloem is responsible for Chapandi. Phloem is responsible for what? Phloem is responsible for transporting food material from leaves to other parts of the plant. So this is the function of xylem as well as phloem. Clear, Nama? So xylem and phloem together constitute vascular bundles. Xylem, phloem renditni kalipite emantam manam, we call it as vascular bundles. So based on the presence of cambium, so vascular bundles are of two types. What is meant by cambium, ma? Cambium means what? A separating layer of cells which separates xylem and phloem is called as cambium. So xylem ni phloem ni separate చేసే ఒక సెల్ లేయర్స్ ఉంటాయి, వాటిని మనం cambium అంటాం. So cambium may be present in some plants, cambium may not be present in some plants. If cambium is present, secondary xylem and secondary phloem is also formed. If cambium is absent, you will have only primary xylem as well as primary phloem. That's it. Clear, Nama? Now see in the figure he is seeing here. See, ma, this is the structure which is given. So you are able to see the pith, no? Pith you are able to see, no, which is at the middle. Then you are able to see the outermost structure which is called as epidermis. Clear? Then you are able to see a blue color region which is called phloem. Clear, Nama? Then the surrounding layer is called as cortex, and you are able to see a pink color region which is called as xylem. And in between that phloem and xylem, you are seeing a separation, no, ma? Separation is there, no? Which separation? This separation, this one. Okay, so that separation is nothing but cambium. Are you able to understand? So then in between these two layers, again you will find another structure, another group of cells which is called as intervascular parenchyma, and this total combination of xylem and phloem we can call it as vascular bundles. Clear, no? So this is the overview of vascular bundle system. Clear, Nama? That's it. So therefore, based on the presence or absence of cambium, vascular bundles we can divide into how many types? We can divide them into two types. Open system, and the second one is called as closed system. Are you able to understand, ma? So whether cambium is present or absent, based on that we are dividing this vascular system into two types. Open system and closed system. So here is there anything which we have to see? Okay, ma? So see the structures, ma? So see the arrangement of xylem and phloem. Xylem is presented towards the center, phloem is presented towards the periphery, towards the outside. And both of them you can call it as vascular bundles or vascular tissues. And surrounding this vascular tissues you have ground tissues. And surrounding this ground tissues we have dermal tissues or epidermal tissues. Clear, Nama? That's it. Now let us see what is meant by open type of system and closed type of system. Clear, Nama? So open type vascular bundle. So in this the cambium is present. Okay, ma? Cambium is present between phloem and xylem. So vascular bundles can form secondary xylem and secondary phloem. So this is one important point. Already we discussed many times now. Clear, Nama? If cambium is present, then that type of vascular bundle, what you can call it as open type. Clear, Nama? And when cambium is present, always you can form secondary xylem and secondary phloem. Because these cambium cells are there, Nama, these cells only they will differentiate. From these cells only, cambium cells only, secondary xylem and secondary phloem will be created. And they are called as open type. Then what is meant by closed type? Closed type means cambium is absent. There is no concept of cambium. So here they do not form secondary tissues. Are you able to understand, ma? And here examples he gave for open type of system, which is the example, ma, dicotyledonous stems, and for closed type of system the examples are monocotyledonous type of stems. So in dicotyledons, cambium is present or absent? Dicotyledons, cambium is present. So open type or closed type? Open type. In monocotyledons, cambium is absent. So closed type or open type? So they are closed type. That's it, Nama. So here in the figure he has given very clearly here. So this is the open vascular bundle, ma. Open vascular bundle means what? Cambium is present. And closed type of system means cambium is absent. That's it. Clear, no? And what is meant by cambium? So here you are seeing a small layer of cells, no? So this small layer of cells is nothing but cambium. And that cambium is absent in monocotyledonous type of stems, which is also called as closed type. Okay? Clear, Nama? So based on the arrangement of xylem and phloem, again vascular bundles we can divide them into two types. Clear, Nama? So how this xylem and phloem they are arranged? So based on that, again we are dividing the vascular tissue system into two types. Which is the first one? First one is called as radial type. Clear, Nama? Radial type. So in radial type, what is the arrangement? Let us see. So xylem and phloem they are arranged in an alternate manner on a different radii. Can you say what is the meaning of this one? So xylem is not separated by phloem by a cambium. So they are present alternately. It means first you will find xylem, then at a different radius you will find the phloem. Then you will find xylem, then at a different radius you will find a phloem. So alternate arrangement. So this type of alternate arrangement is called as radial type. And where you see this radial type of vascular bundles, ma, you can see in roots. So this is another important bit which you should remember. And see the diagram of this one. So see, ma, first you will find this xylem, then at a different radius you will find the phloem. Clear, no? Then you will find a xylem, then at a different radius you will find a phloem. Oh, okay, are you able to understand, ma? So if this type of arrangement is present, then it is called as vascular system you can find in roots. Okay. So next one is conjoint type. So first one is radial, second one is called as conjoint type. So xylem and phloem are jointly situated at the same radius of vascular bundles. Are you able to understand, ma? They are present at the same region and at the same radius. So they are seen in stems as well as leaves. So this is the arrangement of conjoint type. So both of them they are conjoint type. One is conjoint closed and the second one is conjoint open. Conjoint open means what? cambium is present. Conjunct closed means what? Cambium is absent. That's it. Clear now? And this conjunct type where you will see in the case of stems as well as leaves. And roots, what you see? In roots you will see radial type. Clear now? So conjunct vascular bundles usually have phloem located only on the outer side of the xylem. Usually located. Usually located means what? Towards the pith, which one is present? Pith వైపుకి ఏది ఉంటుంది? Towards the pith, xylem will be present. And towards the periphery, which one will be present? Towards the periphery, phloem will be present. Usually. Usually means what? In most of the plants. It means that the opposite is also possible. Are you able to understand, ma? That's it. So therefore, phloem is located on the towards the periphery, and towards the pith, which one is located, ma? Xylem is located. Clear? So let us make a note of that one. So what is the arrangement of xylem? Xylem is towards the pith generally. Okay, ma? And what is the arrangement of phloem, ma? It is towards the epidermis, outermost structure. Clear? So that is one important point which you should remember. Okay? So then we are studying about the anatomy of dicot as well as monocot plants. Now let us. Clear, ma? So first of all we are discussing about the structure of the anatomy of dicotyledonous root. Okay? So transverse section of sunflower root shows the following tissue organization. Okay, ma? If you cut the root of this sunflower plant, then you can find this type of structures which are present. What are the structures? Epidermis, which is also called as epiblema. Clear, ma? Epidermis means what? Total outermost structure is called as epidermis. Clear? And the next innermost structure is called as cortex. And the next innermost structure is called as endodermis. Then the next innermost structure is called as stele. Clear, ma? So inside epidermis what is present? Cortex. Inside cortex what is present? Endodermis. Inside endodermis what is present? Stele is present. Okay. Now let us see the structure here. Clear, ma? So first you can find this pith. Where is this arrangement of this pith, ma? Pith is present in stele. Okay. Then surrounding the pith you will have this vascular bundles, which is a combination of xylem as well as phloem. And beyond that one you can find this pericycle. Okay, ma? This pericycle. Okay. And inside this pericycle only you can find the pith. This is called as pericycle. Inside the pericycle you can find the pith. Are you able to understand, ma? And here only you can find these layers of protoxylem as well as metaxylem, and that is again surrounded by phloem. Okay, ma? Then the structure which is outside this pericycle, it is called as endodermis, and the structure which is above the endodermis or outside the endodermis is called as cortex. And surrounding this cortex you will find the last layer which is called as epidermis, and on the epidermis you will find epidermal appendages which is called as root hairs and trichomes. Are you able to understand, ma? So this is the arrangement of the dicotyledonous stem. Sorry, stem or root, dicotyledonous root. Are you able to understand, ma? So what are the different parts you can see? Epidermis, cortex, endodermis and stele. Now in each of them again which structures are present, we will study each of them in detail. Clear? So see. Okay, ma? So first one we are studying about epidermis, which is also called as epiblema. So it is the outermost layer, ma? Clear? So many epiblema cells protrude as unicellular root hairs. Are you able to understand, ma? So the cells which are present on this epiblema or epidermis, so from them you can find some epidermal appendages which are coming out, no? And these epidermal appendages you can call them as root hairs. For the roots you can find some hairs also which are present, no? And what is the function of these root hairs, can you say? Already we studied previously. What is the function of root hairs? Roots ki malli hairs untai. Clear, no, ma? For example, we have studied taproot system and fibrous root system, no, ma? For a taproot system, again you will find root hairs. So what is the function of these root hairs? Huh? They will increase the surface area of the roots. That's what we have studied. No? They will increase the surface area of the roots. So that if surface area is greater, they can absorb more and more amount of water. That's what we have studied. No? Clear, no? So epidermis is the outermost layer, and most of the cells, they will come out as unicellular root hairs. So unicellular is an important point here. So root hairs are not multicellular, root hairs are unicellular. Clear, no? Next, cortex. Next, which is the layer which is present below the epidermis? Epidermis lopal unnadedi cortex. Clear, no? So cortex consists of several layers of thin walled parenchymatous cells with intercellular spaces. Okay, ma? See, ma, in the figure here you can see, no? This is the cortex. And cortex is made up of what kind of cells? Cortex is made up of parenchymatous cells. Clear, no? And there are some gaps also which are present between one cell and the other cell. So in the figure also you can see some gaps. You are able to see, no, ma? Cells madhyalo gaps unnaya leva? Gaps are present. Are you able to understand, ma? So that is the nature of cortex. So therefore, several layers of thin walled. Walls of these cortex cells are thin, and they are made up of parenchymatous cells, and intercellular spaces are also present. Clear, no, ma? So then we are studying about dicotyledonous roots. Clear? Sorry, in the same dicotyledonous roots we are studying about endodermis. What is meant by endodermis? The layer which is present inside the cortex is called as endodermis. Clear, ma? So it is the innermost layer. It comprises a single layer of barrel shaped cells without intercellular spaces. Okay, ma? So endodermis does not have any type of intercellular spaces. So which is meant by endodermis here? So this structure which you are seeing here, no, ma? So this total layer we can call it as endodermis. Are you able to understand, ma? And this total endodermis, it is made up of which kind of cells, ma? Barrel shaped cells. So you should have seen barrels, no? In which wine will be filled or rum will be filled. Barrels undevi kada pathakalam. So these cells they are having the shape of barrels, barrel shaped cells, and without intercellular spaces. They doesn't have any kind of spaces between them. So the tangential and radial walls of the endodermal cells have a deposition of suberin, so water impermeable waxy material in the form of Casparian strips. So this is another important point which you should remember. Are you able to understand, ma? So these endodermal cells on the tangential sides as well as on the radial walls. Okay, ma? Tangential positions means what? Those positions where one cell is touching with another, that is called as a tangential region. So in the tangential region as well as in the radial walls, what you find? You will find a deposition of suberin. And what is this suberin, ma? So this is a water impermeable membrane or water impermeable layer. Impermeable means water cannot enter into this layer. Water cannot enter into this suberin. That is called as water impermeable. Clear, no, ma? So it is water impermeable and waxy material in the form of Casparian strips. So what is the shape of arrangement of this suberin, ma? It is arranged in the form of Casparian strips. Okay. So are you able to understand what is happening? So this is the endodermis, ma. So barrel shaped cells కనిపిస్తా ఉన్నాయి కదా అక్కడ మీకు. Clear, no? So in between these cells you can find this material which is called as suberin, which is water impermeable and it is a waxy material. Okay, that's it. So then we are studying about the innermost structure which is called as stele. Stele. Clear, no, ma? So all tissues on the inner side of endodermis together constitute the stele. So what is meant by stele? So you will find a layer of endodermis, no? Barrel shaped cells layer is there, no, ma? So inside the endodermis, whatever cells they are present, they are called as stele. And what does a stele consist of? Stele consists of xylem, stele consists of phloem, stele consists of pith. So all these structures they are present inside the stele. Okay, ma? So therefore this stele consists of what? Again it contains the pericycle, it contains the pith, it contains the conjunctive tissue, and it consists the vascular bundles. Are you able to understand what is meant by conjunctive tissue? The cells which are surrounding these vascular bundles, they are called as conjunctive tissues. Clear, Nama? So steel consists of how many parts? Steel consists of pericycle, pith, conjunctive tissue and vascular bundles. Clear? Now we will study each of them in detail. Okay, ma? So first of all, pericycle. Okay, ma? Let us see what is meant by pericycle. So a few layers of thick walled parenchymatous cells. So pericycle is also made up of what kind of cells, ma? Parenchymatous cells. Clear, ma? So these simple cells, they are simple cells only, no? Parenchyma, collenchyma and sclerenchyma, they are simple cells. So where these cells are present, question can be asked on this one also. సో పారెంకైమటస్ సెల్స్ ఎక్కడెక్కడ ఉంటాయి, 4 ఆప్షన్లు ఇచ్చి ఏదో ఒక ఆప్షన్ చూస్ చేసుకోండి అంటారు. అర్థమవుతుంది కదా? సో యూ షుడ్ బి వెరీ కేర్ఫుల్ అబౌట్ ద అరేంజ్మెంట్ ఆఫ్ దీస్ సెల్స్, వేర్ దీస్ సెల్స్ ఆర్ ప్రెసెంట్. క్లియర్? సో పెరిసైకిల్ is made up of what? Thick walled parenchymatous cells next to endodermis. It means immediately inside the endodermis you will find pericycle. So initiation of lateral roots and vascular cambium during secondary growth takes place in these cells, which is a very important point. Okay, ma? So what is the function of pericycle, ma? Pericycle function చెప్పండి. So the lateral roots and vascular cambium, so both of them they start from the pericycle. అర్థమవుతుంది కదా? Roots, lateral roots as well as vascular cambium is developed in the pericycle. Now let us see the structure of this arrangement of this pericycle, ma. So these cells you are seeing here, no? These cells. So these cells inside the endodermis, they are called as what? They are called as pericycle. Are you able to understand, ma? So vascular endodermis local thin layer undi kada, that layer you can call it as pericycle. And from the pericycle, which cells will come out? Lateral roots as well as vascular cambium is developed in the pericycle. Pith. Next, what is meant by pith? It is the innermost region. It is small or inconspicuous. What is meant by inconspicuous, ma? It means it is undifferentiated. Undifferentiated means you don't find a difference between the cells of pith and the surrounding cells. Difference చాలా తక్కువగా ఉంటుంది. It is very tough to identify the location of the pith. That is called as inconspicuous. Are you able to understand, ma? And already we have studied what is meant by pith, no? It may be soft. Are you able to understand? It may be spongy and it may be used for storing starch also. సో కొన్ని చెట్లు మధ్యలో నరికినప్పుడు మీకు లోపల ఒక సాఫ్ట్ టిష్యూ కనిపిస్తుంది చూడండి, మధ్యలో సెంటర్లో. దట్ సాఫ్ట్ టిష్యూ ఇస్ నథింగ్ బట్ పిత్. క్లియర్, మా? దట్స్ ఇట్. నెక్స్ట్, కంజంక్టివ్ టిష్యూ. ఓకే, మా? So the parenchymatous cells, so these cells are also made up of parenchymatous cells which lie between xylem and the phloem. It means xylem and phloem, they are completely surrounded by which cells, ma? Conjunctive cells or conjunctive tissues. అర్థమవుతున్నాయి కదా? So xylem and phloem are present, which are called vascular bundles. So surrounding these vascular bundles there is another tissue which is called as conjunctive tissue. Already I said, you know. Clear, nama? So which is the xylem and which is the phloem here? So these structures they form the xylem as well as the phloem. Okay, ma? And here you are seeing this these cells you are seeing, nama, here. Let me use which color? I will use this color. Clear, nama? So here you are seeing these cells, no? What are these cells? These cells are nothing but the tissues made up of conjunctive tissues. Are you able to understand, ma? So total these vascular bundles of xylem and phloem, they are surrounded by conjunctive tissues. You can remember like this. Clear? Next we are studying about vascular bundles. We know what is meant by vascular bundles, no? So the combination of xylem and phloem is called as vascular bundles. So 2 to 4 xylem and phloem patches may be present. Later a cambium ring develops between xylem as well as phloem. So we are studying in the case of dicotyledonous roots, no, ma? In monocots it is a closed type. Closed type means what? There you don't find any cambium. We are studying this total concept in the case of dicotyledonous roots only, no? And inside the dicotyledonous roots what you find? You will find definitely a cambium will be present. Clear, no? So that is about the steel. Okay. these are the different types of vascular bundles, which I have noted just for your information. So this is open type and this is the closed type. Only these two are mentioned in our syllabus. Clear? So there are other types of vascular bundles also, which is called as bicollateral, so hydrocentric, leptocentric. These they are not present in our syllabus. Clear now? That's it. Okay, so here you can see the conjunctive tissues. Clear? So where this conjunctive tissue is present, conjunctive tissue is present here in the regions of xylem and phloem. You can find these conjunctive tissues. Clear now? That's it. So then now we are studying about monocotyledonous roots. Okay, so in monocotyledonous roots, are they open type of vascular system or closed type? Closed type of vascular system is present. Cambium, is it present or absent? Cambium is absent. Clear now? Now see. So if you see the transverse section of this monocotyledonous roots, then these are the structures which you see. So you will find epidermis, outermost layer, cortex, the next innermost layer, endodermis, the next innermost layer, pericycle, vascular bundles and pith. Same story is again present here also. Clear now? So there are usually more than 6 xylem bundles. So how many xylem bundles are there, Usually 6 bundles. 6 bundles of, bundles means what? Packing. Each packing you can call it as a bundle. Clear now? So 6 xylem bundles are present. Pith is large and well developed. This is one important point. Clear now? So pith is inconspicuous in the case of what? In the case of dicotyledonous stems. In dicotyledonous stems, pith was inconspicuous. What is meant by inconspicuous? Undifferentiated. You cannot differentiate it, you cannot find it very easily. Okay, but in the case of monocotylous stems, pith is highly developed and it is not inconspicuous. It means here you can differentiate it very easily. Are you able to understand? And its size is also quite big size. Okay, and no any secondary growth. What is meant by no secondary growth? There is no concept of secondary xylem. There is no concept of secondary phloem. That is called as no secondary growth. Are you able to understand? If cambium is present, then only you can find a secondary xylem and secondary phloem. Cambium is absent, no? So there is no secondary growth here. Clear, no? That's it. And now this is the transverse section of monocot roots. Clear, no? So metaxylem, the xylem which is present towards the inner side of the pith, and then you will find the innermost region which is called as pith. Then you can find the protoxylem, outermost the xylem which is present towards the periphery, that is called as protoxylem here in this condition, and the next structure is called as phloem. Then you can find the pericycle inside the endodermis, and endodermis you can find inside the cortex, and cortex you can find inside the epidermis, and on the epidermis we have the what you call epidermal appendages, which is called root hairs. Clear, Nama? That's it. Okay. So next we are discussing about dicotyledonous stems. Clear? Until now we have studied about roots, Nama. Now we are discussing about the stems, dicotyledonous stems. Clear? So in a dicotyledonous stems, what you find, basically you will find 2 layers, epidermal layers as well as cortex. Clear, no? So outermost protective layer is called epidermis, covered with a thin layer of cuticle. It may bear trichomes and few stomata. Clear, no? So what is meant by cuticle, ma? Cuticle ante a waxy layer which is present on the leaves as well as the stems. So leaves paina, stems paina oka wax lanti layer untundi. And what is the use of this cuticle, ma? Use of cuticle is what? It is responsible for stopping of water from transpiration. So water evaporate avakunda ee waxy layer addu padutu untundi anamata. Are you able to understand, ma? But cuticle where you don't find? In the roots you don't find any cuticle. Because from the roots definitely water should be absorbed, no? If cuticle is present, water can never be absorbed. Clear? Because it is a waxy layer. Clear? So that is about epidermis. Cortex. What is meant by cortex? The layer just below the epidermis is called as cortex. So multiple layers of cells are present, arranged in between epidermis and pericycle. So it consists of 3 subzones. So cortex, where it is present, cortex is present in between epidermis as well as pericycle. Are you able to understand? So that is the arrangement of cortex in the case of dicotyledonous stems. So it consists of three subzones. So cortex again consists of three zones. In which region we are studying? We are studying in the case of dicotyledonous stems. అది మర్చిపోకూడదు మీరు. అంతా చదివి లాస్ట్ కు రాముడు ఏమవుతాడు సీత ఏమవుతాడంటే కష్టం. ఫస్ట్ యూ షుడ్ రిమెంబర్ వీఆర్ డిస్కసింగ్ అబౌట్ విచ్ స్ట్రక్చర్. వీఆర్ డిస్కసింగ్ అబౌట్ dicotyledonous stems. Clear Nama? So this cortex again has three layers. What are they? Hypodermis, so cortical layers, and endodermis. Able to understand ma? So cortex is made up of what? Cortex is made up of hypodermis, cortical layers, and endodermis. So all these three you can call it as the cortex. Clear no? That's it. Okay ma? So let us shall we see this transverse section of dicot stemma. So see here. So this is the structure of transverse section of dicot stemma. See ma, here in this condition you can find a very big pith. No? Pith is very large. Are you able to see? The innermost soft region is called a pith. No, ma? It is very big in size. It is highly developed. So medullary rays are present. Clear no, ma? Vascular bundles are present. Pericycle is present. Endodermis is present. Parenchyma layer is present. Hypodermis is present. Epidermis and epidermal hair. So once you go through the structure of this one carefully, then I hope you can understand very easily. Are you able to understand ma? This is the transverse section. Transverse section means what? For example, if I take this root, if I cut this root like this, then if I observe the layer, then this is the structure which I see. Clear no ma? But if I take the longitudinal section of dicot root, longitudinal section, that is if I cut it like this, then no, this is also transverse section only ma. This is not longitudinal section, transverse section only. Clear no? So this is the pith ma. So above the pith what you find? Above the pith you will find protoxylem, and surrounding or above the protoxylem you can find this metaxylem. Then you can find a cambium, a differentiation is there. Clear no ma? Then you will find the phloem. So xylem and phloem, they are separated by cambium. Okay ma? And above the phloem you will find pericycle. Above that one you will find endodermis. Then you can find a layer of parenchymatous cells. Then you can find a layer of collenchyma, and finally epidermis, and above the epidermis you can find the cuticle. Clear no? So this is the arrangement of this dicotyledonous stems. Fine? Okay ma? Now we are studying each of them in detail ma. So see here. Hypodermis. What is meant by hypodermis? Clear ma? So this hypodermis, cortical layer, endodermis, all they belong to what? They belong to cortex only. Clear? So that you should be clear. So outer zone just below the epidermis is called as hypodermis. Clear no ma? The layer just below the epidermis is called as hypodermis. It is a part of cortex. So it consists of a few layers of collenchyma. So very few layers of collenchyma are present. So already we have studied no ma? So this here you are seeing this collenchyma layers no? These layers, the layers of collenchyma. Clear no? So it provides mechanical strength to young stem. So what is the purpose of hypodermis ma? Purpose of hypodermis is providing strength to the plant, mechanical strength to the plant. Next you find cortical layers. Okay ma? So what is meant by cortical layers? It is just below the hypodermis. So they consist of round thin walled parenchymatous cells with conspicuous intercellular spaces. Are you able to understand? So let us see in the figure if it is given ma. Cortical layers are able to see. Cortical layers differentiation he did not give no in this figure. So below the hypodermis you can find this cortical layers ma. So these layers. So you are seeing these layers of parenchyma cells no ma. Okay, green color layer kanipistundi kada meeku. This layer. Okay ma. This layer which is present inside the hypodermis. Clear no? That layer we can call it as the layer of cortical layers. So it is made up of what? It is made up of parenchymatous cells. Clear no ma? Okay. So conspicuous intercellular spaces. What is meant by conspicuous intercellular spaces? It means intercellular spaces you can observe very easily. That is the meaning of this one. Inconspicuous means so you cannot differentiate very easily. Conspicuous means very easily you can find the intercellular spaces in this parenchymatous cells. Clear? That is the cortical layer. And inside the cortical layer again you find another layer which is called as endodermis, and it is the innermost layer ma. So cells are rich in starch grains. So it is called starch sheath. So these cells of endodermis, which is the innermost layer, so they can store a huge amount of food material which is called starch. Clear? So that is why they are also called as starch sheath also. So endodermis, what you call it ma? Endodermis you can call it as starch sheath, where in the case of dicotyledonous stems. Clear? That's it. Okay. So then we are studying about the final structure which is called as stele. So inside the endodermis again what you find? You will find the stele, and in this stele only you will find pericycle, vascular bundles, pith, etc. Clear no ma? So stele consists of what? Stele consists of pericycle, it consists of vascular bundles, that is xylem and phloem, then it contains the medullary rays. What is meant by medullary rays? Already we have discussed no ma? Radial rays. Okay ma? And then you will find the pith. So all these structures they are present inside the stele. Clear? Okay. So then we are discussing about each of them again in detail. Pericycle. So already we know, so we have some idea about pericycle no ma? So it is present on the inner side of the endodermis and above the phloem in the form of semi-lunar patches of sclerenchyma. So this is one important bit which can be asked. So what is the arrangement of this pericycle ma? So pericycle is present above the phloem. First you will find xylem no? Then you will find the phloem. Then above the phloem you can find this pericycle. Are you able to understand? So this pericycle is made up of what? Semi-lunar patches of sclerenchyma. Are you able to understand ma? So it means they are made up of which kind of cells? Sclerenchymatous cells. Are you able to understand? Then we study about medullary rays. What is meant by medullary rays? Few layers of radially placed parenchymatous cells between vascular bundles. In between the vascular bundles, already we have drawn the diagram previously, no ma? So these medullary rays, they will be traveling like this radially. Okay ma? And they are responsible for water transportation also. Clear no? So few layers of radially arranged parenchymatous cells. So medullary rays are made up of what kind of cells? Parenchymatous cells. Clear ma? In between the vascular bundles. Vascular bundles, already we know that. So they are large in number. Their ring arrangement is the characteristic of dicot stem. So each vascular bundle is conjoined and open. Clear ma? So protoxylem is enlarged. So what is the meaning of all this structure, all this what you call vocabulary? Already we have studied previously, no ma? So ring arrangement, they are arranged in the form of rings ma. They are arranged in the form of rings, where in the case of dicot stems. Clear no? So vascular bundle is conjoint. So it is not radial, it is conjoint. What is meant by conjoint? Already we have studied no? So above the xylem, immediately you will find the phloem, separated by cambium or may not be separated by cambium. So this type of arrangement of vascular bundles is called what? Conjoint. Already we have studied no? Arrangement of vascular bundles is two types. One is radial, another one is conjoint. Radial means what? So alternately xylem and phloem they are arranged. Okay ma? Conjoint means above the xylem you will find the phloem. So they are conjoint and open type. Open type means what? Cambium is present. Are you able to understand ma? So protoxylem is enlarged. So this type of arrangement also we have studied no ma? The arrangement of xylem, enlarge and exarch. Okay. So what is meant by enlarge and exarch? Metaxylem, if it is present towards the pith, then it is called as endarch. Antena? Kada? Chudandi osari, I too forgot this one. Metaxylem, 1 minute, let us see this. Endarch, exarch. Endarch means protoxylem towards the pith, no? That's it. Clear, ma? When protoxylem, when it is present towards the pith, then it is called as endarch. Okay. So this is the arrangement of vascular bundles in the case of dicotyledonous stems. Clear, no, ma? Okay. So then pith. What is meant by pith? Already we have understood, no? So the central portion of the stem. So it has many rounded parenchymatous cells with large intercellular spaces. Already we have studied. Clear, no, ma? So then we are studying about monocotyledonous stems. Dicotyledonous stems we have studied, no? Now we are studying about monocotyledonous stems. So hypodermis is made up of sclerenchymatous cells. Hypodermis is made up of what? Sclerenchymatous cells. So scattered vascular bundles. Vascular bundles are scattered. Each surrounded by a sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. Are you able to understand, ma? So in monocot stem, so scattered vascular bundles you can find, and they are surrounded by which kind of cells, ma? They are surrounded by sclerenchymatous cells. So there is a large conspicuous parenchymatous ground tissue is also present. So vascular bundles are conjoined and they are closed. Conjoined means what? For example, if this is the xylem and above the xylem you can find the phloem. That is called conjunctinoma, and it is closed. Closed means you don't find any cambium which is present between them. Already we have studied now in monocotyledonous stems you don't find any cambium. So peripheral vascular bundles are smaller than centrally located ones. What is the meaning of this one? Peripheral means what, ma? So here we already said that this vascular bundles, they are not present in a what you call radial structure. In dicotyledonous stems, they were arranged in a particular fashion, no? Are you able to understand, ma? They were arranged in a radial fashion. Are you able to understand? But in the case of monocot stems, vascular bundles, they are not present in a regular fashion. They are scattered. Scattered means what? They are present throughout the stem wherever they like. That is called as scattered vascular bundles. And these scattered vascular bundles, they are surrounded by which kind of cells? Sclerenchymatous cells. Are you able to understand? So peripheral vascular bundles are smaller than centrally located ones. So this is another important point. Peripheral vascular bundles means what, ma? The vascular bundles which are present towards the outer structure, that is towards the epidermis, they are very small when compared to the vascular bundles which are present towards the pith, towards the center. అంటే మధ్య వైపుకున్న vascular bundles పెద్దగా ఉంటాయి. మీరు ఎడ్జ్కు వెళ్లే కొద్దీ vascular bundles సైజెస్ ఏమైపోతాయి? తగ్గుతూ వెళ్తాయి. అర్థమవుతున్నాయి కదా? So, but in the case of dicot stem, the arrangement of xylem and phloem was regular, no? Regular arrangement was there. That was the property of dicot stem, no? But here it is in the form of scattered pouches. That means what here? So here you can find one scattered pouch. Here you can find one scattered pouch. Here you can find one scattered pouch. Here you can find. So they are arranged in scattered pouches. Are you able to understand? But towards the periphery, so here you can see they are very small in size. Okay, ma? Epidermis వైపుకి ఉన్న ఈ vascular bundles, they will be very small in size, but towards the center they are very big in size. That is the point which you should understand. So the Phloem Parenchyma is absent here. Are you able to understand, ma? So this is another important bit. So Phloem Parenchyma is absent and water containing cavities are present within the vascular bundles. So within the vascular bundles what you find? Cavities. Cavities means what, ma? Gaps. And these gaps, they are responsible for carrying water, just like medullary rays in the case of dicot stems. In dicot stems you will find medullary rays, no? and those medullary rays, they were responsible for carrying water. But here you will find water carrying cavities will be present, which are again responsible for carrying water. Are you able to understand? Ma? So that is about monocot stems. So what is the difference, major difference between monocot stem and dicot stem? There you will find a regular arrangement of vascular bundles in dicot stem. But here you will find these vascular bundles, they are present wherever they like. Clear, Nama? That is one difference. Clear? That's it. So then leaf we will study in the next class. Ma. Dicotyledonous leaf. Okay. We will stop up to here and we will continue in the next class. Sima. So next we are studying about dicotyledonous leaf. Dicotyledonous leaf, it is also called as dorsiventral leaf. Okay. Now if you take the vertical section of this leaf, okay, then it has three main regions. Now what are they? Epidermis, mesophyll and a vascular system. What is meant by epidermis? Outer covering is called as epidermis. And mesophyll. Mesophyll all means already we have studied in the last class, no? So mesophyll is made up of parenchymatous cells which contained thin-walled chloroplast. We have studied, no? So walls of the chloroplast are very thin and they have chlorophyll pigment in them, which is responsible for producing starch with the help of photosynthesis. So this type of cells you can call them as mesophyll cells. Cells which contain chloroplast. Clear, no? And then you will find a vascular system also. Okay. Now if you see in the figure of this dicotyledonous leaf of a dicotyledon plant, then first of all you have adaxial epidermis. What is meant by adaxial epidermis? So epidermis, which is lying on the topmost region, is called as epidermis. That is at the top of the leaf. Okay. And on the adaxial epidermis, again you will find a cuticle. Cuticle means the waxy layer. Here you will find, you are seeing a small green color layer which is there, Nama. So this small green color layer is called as cuticle, which behaves like a wax. Are you able to understand? Record out on the other. Okay. Then what is the bottommost epidermis? It is called as adaxial epidermis. And here you can find the stoma. Stomata you can find at the adaxial epidermis only. Okay, ma? See, ma, stomata you can find on the top of the leaves also, but they are very rare. Very rare, or you can say almost it is zero. Are you able to understand, ma? On the top of the leaf also you can find stomata, but they are very, very, very less. So all the stomata you can find at the bottom of the leaf only. Okay, that is where you will find in the abaxial epidermis only you will find the stomata. Clear? Then we have polysaid mesophils. So mesophils you can divide into two types, polysaid mesophils and spongy mesophils. Clear? So this is polysaid mesophils. So they are like elongated cells. So ikkada cells ela unnayi, they are just like elongated cells, no? So that is the type of cells which are present in polysaid mesophil. Then here you will find spongy mesophil with some air cavities in between them. So in polysaid mesophil you don't find any gaps. Gaps are very less. But in a spongy mesophil you can find gaps are present in between the mesophil cells. Are you able to understand? Then you can find this xylem and phloem, and surrounding this xylem and phloem you will find a sheet of layers which is called as bundle sheath. So here you are seeing this green color structure, no ma? One green color layer you are able to see here, no? Clear? So that layer is nothing but a bundle sheath. There you are able to see, no? So bundle sheath, what it will do, it will surround the xylem as well as phloem. Are you able to understand? So this is the transverse section of this dicot leaf. Okay. So first of all we will study about epidermis. Okay, ma? What about epidermis? So it covers both the upper surface and lower surface. Upper surface epidermis is called as adaxial epidermis, and lower surface epidermis is called as abaxial epidermis. Clear, ma? So it has a conspicuous cuticle. Conspicuous cuticle means what? Cuticle means waxy substance, no ma? Waxy substance which is responsible for stopping transpiration. Already we have studied, no? What is meant by transpiration? Evaporation of water is nothing but transpiration. So waxy substance is present on the leaf which is responsible for stopping the transpiration of water. So adaxial epidermis generally bears more stomata. Clear ma? So adaxial means bottommost epidermis, no ma? The epidermis which is present on the bottom of the leaf or below the leaf. And in adaxial epidermis, that is at the top of the epidermis, stomata are fewer or absent. Stomata are very less or generally they are absent also. Are you able to understand? So this is about epidermis. So this figure already we have studied. Clear, no ma? So mesophyll. So first of all we have studied about epidermis, no? First layer is called as epidermis, then followed by mesophyll. Clear? What is this mesophyll? Already we have studied previously, no ma? It is made up of parenchymatous cells. It consists of a thin-walled chloroplast which is responsible for producing starch with the help of photosynthesis. So tissue between upper and lower epidermis is called as mesophyll. Clear, no? So it is made up of what? It is made up of parenchyma. So they contain chloroplast for photosynthesis. It has two types of cells, polycide parenchyma and spongy parenchyma. In the figure you can see here, Noma. So this is called polycide parenchyma and this here is called as spongy parenchyma. This one. Clear, no? That's it. Okay. Then polycide parenchyma. It is adaxially placed. Okay, ma? So adaxially placed means what? It is present on the top of the leaf, topmost regions of the leaf. And abaxial means it is present on the bottom of the leaf. Clear, no ma? So it is adaxially placed, made up of elongated cells arranged vertically and parallel to each other. Okay. So here in the figure you can see that all these cells they are placed vertically, no ma? They are placed side by side and they are placed vertically. So that is the arrangement of polycide parenchyma. Next, what about the spongy parenchyma? So the oval are round shaped and they are loosely arranged. It is situated below the polycide cells and extends to the lower epidermis. Lower epidermis means abaxial epidermis. So there are numerous large spaces and air cavities between these cells. So therefore mesophyll you can divide into two types, polycide mesophills, spongy mesophills. Polycide mesophills you can find on the top of the leaves, topmost regions of the leaf, whereas spongy mesophyll you can find on the bottom of the leaves. And in polycide parenchyma you don't find any spaces, but in spongy parenchyma you will find some spaces. Clear, Nama? That's it. Okay. So then vascular system. Now we are studying the third system. So first one we have studied about epidermis, no? Abaxial and adaxial. Then we have studied about mesophyll, polycide mesophyll and spongy mesophyll. Now we are studying about the transport system which is called as vascular system. Okay. So vascular system includes what, ma? It includes vascular bundles. Clear, no ma? Vascular bundles are present. So they can be seen in veins as well as midribs. What is meant by veins and midribs? For example, if you take the shape of a leaf, so leaf appears something like this, no ma? So this is the petiole and this is the midrib. Clear, no ma? And midrib is again having some veins. Okay, and veins are again divided into veinlets also. Okay, so therefore vascular system, where you find vascular system you can find in veins as well as midribs. Are you able to understand? Vascular system మీకు veinlets లో కనిపించదు. Veins and midrib only you can find the vascular system. Are you able to understand? That's it. So size of vascular bundles is dependent on size of the veins. It means if size of the veins is bigger, vascular system is also bigger. Clear, Nama? The veins vary in thickness in the reticulate venation of dicot leaves. Okay, ma? So dicot leaves have reticulate venation, no? Dicot leaves have what kind of venation? Reticulate venation. What is meant by reticulate venation? There is a midrib, there is a vein. Vein is again divided into veinlets. So a network of veinlets is created. That is called as a reticulate venation, no? Are you able to understand, ma? So veins vary in thickness. So different veins have different sizes in the case of reticulate venation. Venation already we have studied two types of venation, no ma? One is reticulate venation, another one is parallel venation. This is the example of reticulate venation. Parallel variation means what? A leaf will be like this. So this is the mid. So just you will have all the veinlets either like this or all the veinlets they are arranged either like this. So this type of arrangement is called as parallel venation. So this is reticulate and this is for parallel. Parallel venation. Understood, Amma? That's it. So veins vary in thickness. It means the thickness of the veins is not constant. They are varying continuously. So vascular bundles are surrounded by a layer of thin-walled, thick-walled bundle sheath cells. Already we have studied, no? Vascular system is totally composed of what? Here, around this xylem and phloem, you are seeing a layer of cells, no? Green color cells you are seeing, no? So these cells, they are called as bundle sheath. Bundle sheath of cells, this one. Clear, no? That's it. So then we are studying about monocotyledonous leaf. So previously we have studied about dicotyleaves, no? Now we are studying about monocotyleaves. So the anatomy of monocot leaf is like that of dicot leaf in many ways. So almost monocot and dicot leaves, they appear to be almost similar. Okay. So it shows the following differences. Some differences are there. Stomata, they are present on both surfaces of the epidermis. That is, in the adaxial epidermis also you will find stomata. On the abaxial epidermis also you will find stomata. Stomata you can find on both sides. And mesophyll, it is not differentiated into polycide and sponge. In the case of dicot leaf, it was divided into two types, Nama. Mesophyll was divided into polycide as well as sponge. But here there is no such division. There is only one kind of mesophyll cells. They are not divided into polycide, they are not divided into sponge. Clear, ma? So these are the two important differences in the case of monocot and dicot. Remaining all the characteristics, they remain same. Understood, Nama? So same concept here also. Of adaxial epidermis, xylem, mesophyll, substomatal cavity, abaxial epidermis, stoma, everything remains the same. Clear? That's it. Okay, ma? So in grasses, next we are studying about the same leaf, monocotyledonous leaf only. In grasses, clear, certain adaxial epidermis cells along the veins, they modify into large empty colorless cells. These are called as bulliform cells. Clear, Nama? When they have absorbed water and are turgid, the leaf surface is exposed. And when they are flaccid because of water stress, they make the leaves curl inwards to minimize water loss. Okay. So this is one special characteristic of monocotyledonous leaf. Okay. So here what is happening, ma? Some of the cells belonging to adaxial epidermis. Adaxial means the topmost region or bottom region? Topmost region. So these cells, they have the ability to absorb water and they are called bulliform cells. Are you able to understand? So when these cells, when they absorb a huge amount of water, they spread the leaf so that maximum area is exposed to the atmosphere. Are you able to understand? So that the water which is not necessary, more than required water, that is evaporated. Okay. And when the same cells, when they are a deficit in water, that is when water is less, then what they do, they curl the leaves. They will twist the leaves so that the surface area of the leaf is reduced and the loss of water is reduced. Are you able to understand, ma? So this is the language which is called turgid and flaccid. Turgid means what? Swollen. Huge amount of water when it is present, leaf becomes swollen and total surface area of the leaf is exposed to the atmosphere. అర్థమవుతుంది కదా? Then what is meant by flaccid? Flaccid means ముడుచుకొని పోవడం. When water is very less, then the leaves become flaccid and they will curl the leaves, ముడుచుకొని పోతుంది అనమాట. So that surface area of the leaf is reduced and water evaporation is reduced. Understood, Amma? This is the purpose of these bulliform cells. So here in the figure you can see, ma. So these cells you can call them as bulliform cells. These cells. So they are present inside the below the cuticle. No? Cuticle కింద ఉన్నాయి చూడండి. Are you able to see? So they are present below the cuticle. They are called as bulliform cells. Are you able to understand, ma? That's it. Okay. So next, parallel venation. Parallel venation is reflected in the near similar sizes of vascular bundles except in main veins. So already we studied previously, no, in morphology of plants we have studied, no? Clear, no? Dicot leaves, what is the shape of dicot leaves? What is the type of venation they have? Reticulate venation. But in monocot leaves, they are having parallel venation. Clear, no? So parallel venation is present here, and similar sizes of vascular bundles are present except in main veins. Are you able to understand, ma? It means almost all the vascular bundles, that is the combination of xylem and phloem, they are having similar sizes. Okay, but that was not the case in the case of dicot leaves, no? In dicot leaves, veins, they are varying in sizes. So therefore, vascular bundles also they vary in sizes. అర్థమవుతుంది కదమ్మా? ప్రణీత్ వింటా ఉన్నావా? ఓకేమా? So you will find similar sizes of vascular bundles in monocot leaves, but the vascular bundles are having different sizes in the case of dicot leaves. What is the reason? Size of veins is different in dicot leaves, no ma? Reticulate venation ఉంటుంది కదా అక్కడ, ఒక్కొక్క vein ఒక్కొక్క size లో ఉంటుంది. So therefore, xylem and phloem bundles also they will be having different sizes. Okay, in dicots, but in monocots, all of them you will find the same size. Clear, Nama? So that is about the leaf anatomy in the case of monocot as well as dicot leaves. Okay, then we are studying about the next topic, which is called as secondary growth. Okay, now see the structures which are present here, ma. So phloem is present, xylem is present, scleral chymatous cells they are present. Clear, nama? And here you can find this pith. This is the pith. Okay, ma? So this one is the primary growth. This one belongs to primary growth, and this one belongs to secondary growth. Okay, so pith is common for both the primary as well as secondary growth. Then these cells, this one you can call it as primary xylem. Are you able to understand, ma? And surrounding the primary xylem, you have the secondary xylem is present. Clear? Then you will find a layer of cells on the outermost structure, which is called as cork cambium. Then outermost structure is called as epidermis, and the layer which is next to cortex or inside the epidermis is called as cortex. And this total structure which is growing outside this primary growth, this you can call it as the bark. Clear, Nama? And epidermis is covered by cork. అర్థమవుతుంది కదా? So what are the structures which are present in bark, ma? Bark లో ఏమేమి ఉన్నాయి చెప్పండి? In the bark you find what you find? You will find a cortex, you will find epidermis, you will find cork cambium. So these are the three structures which are present in the bark. Are you able to understand, ma? So cork cambium also belongs to bark only, epidermis also belongs to bark only, cortex also belongs to bark only. Clear? So this structure you can call it as the secondary growth. Then you have the primary phloem. This one is called primary phloem. Then this one is called the secondary phloem. Are you able to understand, ba? So touching the cambium, so secondary phloem and secondary xylem will grow. అర్థమవుతుంది కదా? So previously we have studied open system and closed system. No? What is meant by open system of vascular system? Closed means there is no cambium there. There is no layer of cells in between phloem and xylem. What is meant by closed? Closed means closed or open? Open type means what? So you will find a layer of cambium in between xylem and phloem. So this layer of cambium, it develops into secondary xylem and secondary phloem. Already we have studied, Noma. So that is the indications which is shown in this figure here. Okay. So surrounding the cambium, you will have secondary xylem and secondary phloem. Secondary xylem develops towards the inside. Secondary phloem develops towards the outside, out of cambium. Clear, no? That's it. So that's total study. We will study about secondary growth. Okay, ma? Now see. So the growth of the roots and stems in length with the help of apical meristem is called as primary growth. What is meant by primary growth, ma? So roots and stems, they will grow, no? And their length will increase with the help of apical meristem. Apical meristems, again there are two types, no? Shoot apical meristem, root apical meristem. Shoot apical meristem means The meristems which are responsible for growing on the length of the stems. Shoot apical meristems means the meristems are dividing cells which are responsible for growth of the roots. Sorry, root means. Shoot means which moves upwards. So this primary growth is called primary growth. That's all. Dine primary growth anta. Then what is meant by secondary growth ma? Secondary growth means it is the increase in girth of dicot plants. So what is meant by girth here? This developing of cork, this development of bark, this development of secondary xylem, this development of secondary phloem. This is called as girth. Size పెరుగుతుందన్నమాట. అర్థమవుతుంది కదమ్మా? Around the primary growth, you will find the development of cork, no? You will see the development of bark. Okay, ma? This total growth we can call it as secondary growth. అర్థమవుతుందా? Primary growth కి secondary growth కి difference అర్థమవుతుంది కదమ్మా? Primary growth means just shoot apical meristem and root apical meristem. Stems they grow in length and roots they grow in length. And around the stems you will find another layer which is called a secondary layer, and that growth is called secondary growth. Okay, now see, so here in secondary growth again we can divide this one into two types. What is the first one? Vascular cambium, and the second one is called as cork cambium. Vascular cambium means what? Secondary phloem, secondary xylem. When they develop, then it is called as vascular cambium in the case of secondary growth. Okay, ma? And cork will develop, no? Cork also will develop, no ma? So outside the plant, so tree, you can find dead cells, no ma? చెట్టుకు అదేమంటారు, దాన్ని ఇది ఇంకా తెలుగులో ఏమంటారు, బార్క్ ఉంటుంది కదా. బార్క్ అంటే ఇదంతా కలిపి బార్క్ అవుతుంది. పైన డెడ్ సెల్స్ ఉంటాయి కదా. సో డెడ్ సెల్స్ యూ కెన్ కాల్ ఇట్ యాజ్ కార్క్ cambium. Okay ma? That's it. So secondary growth you can divide into two types. One is vascular cambium and the second one is called as cork cambium. Now we will study each of them in detail once again. So see here. Okay ma? First let us see the different parts which are present ma. Then we will try to understand the theory part here. Okay? So secondary growth in a dicot stem, stages in transverse views. Clear no? So see here. So this one is the primary growth ma, and this one is the secondary growth. Okay, now see the structures in the primary growth. First one, pith is present. Okay ma? Then you have primary xylem, then you have vascular cambium which is present in between xylem and phloem. Then you have primary phloem, then you will find a cortex, then you will find the epidermis. These structures already we know, no? There is nothing new here. Okay ma? Then coming to secondary growth. Clear no ma? So cambium ring is present. Okay, secondary phloem has developed, secondary xylem has developed, and medullary rays also they have developed. Then a layer of cells called phloem has developed. Clear no ma? So this is about secondary growth. Are you able to understand? So secondary growth is of two types, no ma? Vascular growth as well as cork growth. Clear? Now let us see. So vascular cambium, secondary growth. In secondary growth we are studying this vascular cambium. So it is the meristematic layer responsible for cutting of vascular tissues. Cutting of vascular tissues means what ma? Separating the vascular tissues. Okay ma? Xylem and phloem. So xylem and phloem, they are separated by vascular cambium only, no? So it separates the xylem and it separates the phloem. So in the young stem, it is present in patches as a single layer between xylem and phloem. Later it forms a complete ring. Okay, what is the meaning of this schema? So primary growth you are seeing, no? This primary growth is nothing but young stem. Young stem అంటే పూర్తిగా స్టెమ్ డెవలప్ అవ్వకముందు దాన్ని ఏమంటాం మనము? We call it as young stem. So in young stem you are seeing this green color discontinuous lines you are seeing, no? So these green color discontinuous lines they are called as cambium. And previously they were in patches. Patches means what? They were not continuous. But when this primary stem, when it grows into mature stem, then this ring, this discontinuous patches, all of them they get connected and a circular ring will be formed. Are you able to understand ma? So I will try to draw the diagram here. So in primary stem, this cambium, it will be like this, discontinuous. Are you able to understand? Ma? But when the stem matures, what happens? They will complete into a ring. So that is the point which is trying to explain us. Are you able to understand? So that is what is happening. So in young stem, it is present in patches in only one layer. But when the stem matures, they will form a complete ring. A ring of cambium will be formed. Clear, Nama? Okay. So that's what you can see in the figure, no? So here the cambium ring is a complete one. Okay. But here it is present, cambium, vascular cambium is present in only patches. Fine. That's it. So formation of cambial ring. So formation of cambial ring also, it will come under secondary growth only, no? Are you able to understand? Ma? In primary stem, it was in patches. It was a discontinuous. But here, when cambium ring, when it completes into a ring, then it is called a secondary growth. Clear? Now see. So in a dicot stems, cells of cambium present between primary xylem and primary phloem is intrafascicular cambium. So what is the name of this cambium? Ma? So this name of the cambium is intrafascicular cambium. Are you able to understand my dicot stems? So cells of medullary race. What is meant by medullary race? Already we have studied, Nama. So these rays, you can call them as medullary rays. Are you able to understand? These are medullary rays. So cells of medullary rays adjoining this interfascicular cambium become meristematic and form intrafascicular, no, first one. This one is interfascicular cambium. Thus, a continuous ring of cambium is created. Are you able to understand? So, cambium previously it was present only between xylem and phloem, noma. Then that kind of cambium, what you call, you can call it as intrafascicular cambium. Intrafascicular. Intrafascicular in which condition? When it was present in the form of patches. First two primary stem lo patches మాత్రమే ఉన్నాయి కదా. So that cambium you can call it as intrafascicular cambium. Then after the cambium ring it is completed. It is completed because of interfascicular cambium and then a continuous ring will be created. Are you able to understand? It means in between two intrafascicular cambium, which cambium is present? Interfascicular cambium will be present. అర్థమవుతుందా? Are you able to understand, ma? Not able to understand. See, I will try to draw the diagram here. See, ma. So initially, cambium was present in patches, no? Cambium was present in patches. Now, what is this cambium called as? Intrafascicular cambium. Understood, Nama? And these gaps, in these gaps, what you find? In these gaps, you can find the medullary rays. Clear, no? And this medullary rays, they will make another cambium which is called as interfascicular cambium to be present. Clear, Nama? These gaps are filled by what? Gaps are filled by interfascicular cambium. So therefore, it completes a ring, no? That's what he is trying us make us to understand. Are you able to understand? In between two intrafascicular cambium, what you find? You will find a interfascicular cambium. In between two interfascicular cambium, what you find? You will find a intrafascicular cambium. So that is how from the primary cambium, secondary cambium of ring is created. Are you able to understand? So that is the construction which is happening here. Clear, Nama? Okay. Now let us see what is the activity of this cambial ring. What is the functioning of this cambial ring? Clear, Nama? Now see. So the cambial ring becomes active and cut off new cells. Okay, ma? So this cambium ring, it becomes active and it starts dividing. It starts dividing. It divides into what? On the outside it divides into secondary phloem. On the inner side it divides into secondary xylem. That is what will happen. Clear, Nama? So the cambial ring becomes active and cut off new cells, both towards the inner and outer sides. The cells cut off towards the pith. Pith means what? Inner side. Mature into secondary xylem. Already we discussed. No? The cells cut off towards the periphery, mature into secondary phloem. అర్థమవుతుంది కదమ్మా? So previously what was happening previously? See, I will try to understand here. So consider this is a vascular cambium. This is the vascular cambium. Okay. So this one is what? This one is primary xylem. Clear, ma? Primary xylem. And what is this one? This is primary phloem. Okay, and here you will find pith. This is what you find, center of the stem, which is called as pith. Now this cambium, this is the cambium, no? This cambium, it divides continuously. Divides continuously. Are you able to understand, ma? And here another layer is created, which is called as secondary xylem. Secondary xylem. And here, let us draw with another color, ma. So this one. This one is called secondary xylem. And here you will find another layer, which is called as secondary phloem. Are you able to understand? Clear? So this is about the functioning of this cambium. అర్థమవుతుంది కదా? So this cambium divides continuously. On the inside, what is formed? A secondary xylem will be formed towards the center, towards the pith, and towards the periphery or towards the outer surface, secondary phloem will be created. Clear, Nama? So this is the function of this cambium ring. Okay. So cambium is more active on the inner side than on the outer side. This is another important point. Clear, ma? So cambium definitely divides, but it divides more on which side? It divides more on the inner side, that is towards the xylem side, and it is less active towards the phloem side, outside. Clear, no? As a result, more secondary xylem is produced than secondary phloem, and soon forms a compact mass. So అర్థమవుతుంది కదా ఏం జరుగుతుందో? So cambium doesn't divide equally on both sides, ma. It is more active on the xylem side. It is less active on the phloem side. So automatically more amount of xylem will be created, less amount of secondary phloem will be created. So this is the function of cambium. Clear? So next. So primary and secondary phloems get gradually crushed due to the continued formation and accumulation of secondary xylem. However, primary xylem remains intact in or around the center. So this is another important point which you should remember. Clear, ma? See, ma, what is happening? Growth is happening more on the inside, no? Secondary xylem is formed very large in mass when compared to secondary phloem. So size of the stem increases from the inside. Growth ekkadi nunchi perugutundi? Lopal nunchi perugutu vastundi. So therefore, when growth continuously happens, more and more pressure will be created on the phloem, no? Which is formed on the outside, and there is no pressure which is created on the xylem. Total pressure falls on the phloem itself. Are you able to understand, ma? So secondary phloem and primary phloem, they get crushed with each other. అర్థమవుతుంది కదా? Compress అవ్వడం స్టార్ట్ అవుతాయి. Clear? At some places, cambium forms a narrow band of parenchyma which passes through the secondary xylem and the secondary phloem in radial directions. These are called secondary medullary rays. This already we have studied, no? What is meant by medullary rays, ma? Medullary rays means we know. So these are radial rays which are made up of parenchymatous cells which are arranged in radial fashion, which are responsible for transporting of water. And here what is forming here? Secondary medullary rays are forming. Okay, ma? And these secondary medullary rays, they are starting from where? They are starting from the cambium. They are starting from the cambium, and they are also made up of parenchymatous cells, and which passes through secondary xylem and secondary phloem. They will pass through secondary xylem also, that is towards the inside also, and they will pass through the outside also, which is secondary phloem also. Are you able to understand, ma? Medullary rays are also, we are dividing into two types now: primary medullary rays and secondary medullary rays. Okay, that's it. So here you can see. Okay, ma? See, ma, see this figure. So what are these, ma? What are these medullary rays? Can you say? Primary medullary rays. They are called as primary medullary rays. And what are these? Secondary medullary rays. Secondary medullary rays will cut which layers? Only secondary xylem and secondary phloem. But primary medullary rays, they start from the pith itself. Pith is not differentiated. Beyond the pith, they will travel up to the end of this stem. Are you able to understand, ma? That's it. Okay, అర్థమవుతున్నాయి కదా? So primary medullary rays and secondary medullary rays both are created in the stem. Okay, Then spring wood and autumn wood. Okay, ma? Wood you can divide into two types, ma. Spring wood and autumn wood. The wood which is developed in the spring season is called spring wood, and the wood which is developed in autumn season is called as autumn wood. Clear? Now see. So here you can, in the figure you can see, ma. Spring wood is light in color, no? What is the color of spring wood? See the figure. Spring wood color ela undi cheppandi? Light color. But if you see this dark color rings, they are called as autumn wood. Are you able to understand, ma? So that is the difference. Now see. So many physiological and environmental factors control the activity of cambium. Clear, ma? So it means the activity of the work of cambium, it is dependent on many factors. Physiological factors are also responsible. Environmental factors are also responsible. In spring season, cambium is very active. So this is one important point. In which season cambium is active, ma? In spring season, cambium is active and produces many xylem elements having vessels with wider cavities. This wood is called as spring wood, or you can call it as early wood. Okay, ma? It is lighter in color and has lower density. Okay. So huge number of divisions will take place in which season? In spring season. So therefore, very light colored wood will be developed by large mass in spring season. So that is the growth of a plant is larger in which season? In spring season, plant grows by huge mass. Clear, no, ma? Whereas in winter season, cambium is less active and forms lesser xylem elements having narrow vessels. This wood is called autumn wood. Clear, no, ma? Or you can call it as a late wood. So that is in autumn season, the growth of plant is limited. Are you able to understand? Whereas in spring season, plant grows by huge mass. That's what he is trying to say. So it is a darker and has a higher density. So therefore, what you see, ma? For example, if this is the total stem, 1 minute. Very good. Clear, no, ma? If this is the total stem, ma, you can see this brown color layers. Okay. So these brown color layers are formed in which season? Autumn season, because the growth of food is not so much. So it grows by thickness by only small amount, and the cells are very tough. Are you able to understand, ma? And the density of these layers is very large. Okay. And what is the color? Color is very dark in color. So these two kinds of food that appears as alternate concentric rings constitute an annual ring. What is meant by annual ring? One dark layer and one light layer. They are formed in 1 year, no, ma? ఒక సంవత్సరంలో ఒక ఆటమ్ సీజన్ ఒక స్ప్రింగ్ సీజన్ వస్తుంది కదా. It means one dark color layer, one light color layer. If you measure, if you count them, it will become 1 year. So depending on how many rings are formed, very easily you can estimate the number of years of the plant. Are you able to understand, ma? If there are 10 rings, 10 dark rings, you can say the life of this tree is, the age of this tree is 10 years. Like this you can say. That is called as annual ring. Okay, ma? So this is used to estimate the age of the tree, which is called as dendrochronology. So what is the name of this science, ma, which is useful for aging of the trees? Dendrochronology. Clear? That's it. So very simple, just by counting the number of rings which are present. Now rings means which rings you have to count? Either dark or either brown. One dark plus one light color, it will give 1 year. So just if you count the number of dark bands, then you can find the number of the years. That's it. Clear? Okay, ma? Next, heartwood and sapwood. Clear, no, ma? So this is also related to vascular cambium. So see where you will find this heartwood and sapwood, ma? So this one. So this total structure, okay, ma? This thick brown color layer you are seeing, no? This I can call it as heartwood. Okay, ma? And the remaining layer, comparatively light colored layer, this I can call it as sapwood. So when you cut a mature stem into two transfer section, then you can see like this, no, ma? At the center you will find a very dark color wood. That is called as heartwood. And surrounding this one you will have a comparatively lighter color wood, which is called as sapwood. Clear, no? Now let us see. So heartwood is the hard dead, dark brown colored, highly lignified and non-functional central part of the secondary xylem of old trees. It means all these cells, they are dead cells. Okay ma? So మధ్యలో ఒక డార్క్ లేయర్ ఉంటుంది కదా, all the cells they are dead cells. Highly lignified, it means a huge amount of lignin is present. Non-functional, dead cells that doesn't perform any function, no? So non-functional. So dark color is due to deposition of organic compounds, tannins, resins, oils, gums, aromatic substances, essential oils, etc. Okay ma? So what is the reason for this dark color ma? Cells are not dark in color, but in this dark region you will find a deposition of many substances. What are the substances which are deposited? Tannins, resins, oils, gums, aromatic substances and essential oils. These are present. These substances make it hard, durable and resistant to the attacks of microorganisms and insects. Okay ma? So చెదలు పడతాయి కదా చెక్కకు. సో ఇట్ ఈస్ చెదలు మనకు సాప్ వుడ్ కు పడతాయి కానీ హార్డ్ వుడ్ కు చెదలు పట్టాలంటే అంత ఈజీ కాదు. బికాస్ ఇట్స్ డెన్సిటీ is very 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 large because of deposition of all these layers. Okay ma? So for the protecting itself from the attacks of microorganisms and insects, so this hardwood can protect itself because of deposition of all these substances. Clear nama? That's it. So sapwood is the peripheral region of the secondary xylem. So where is the positioning of this sapwood ma? Peripheral region means outside region to secondary xylem. Are you able to understand? You know secondary xylem nama? Beyond the secondary xylem, all that wood you can call it as sapwood. Clear nama? And it is living wood and it is lighter in color. It means these cells, they have life. Cells are living and they are lighter in color. Clear? Function. So it is involved in the conduction of water and minerals from root to leaves. From root to leaves, they are responsible for giving water. Clear? So next, cork cambium. So what is meant by cork cambium? We know nama? See here. So this is the heartwood, dark color dead region, and this is the sapwood, light color living region. And this sapwood again we are dividing into many parts. What are they? First you will find a green color region nama, which is called as the combination of xylem and phloem, or you can call it as vascular cambium. Okay. Then you will find bark, living phloem is present. Okay ma? So next, periderm, which contains of cork cambium, or you can call it as the cork. Okay. Now we will study each of them in detail. So as the stem continues to grow in girth due to activity of vascular cambium, the outer cortical and epidermis layers get broken. Are you able to understand ma? So inside the stem what happens ma? Secondary xylem and secondary phloem, they start developing no? But growth is more in which region? Growth is more in secondary xylem. So what happens to secondary phloem and secondary primary phloem? They get compressed, no? And these compressions, they will again exert pressure on the outer layers. So first who will exert pressure? Xylem will exert pressure on phloem. Phloem starts exerting pressure on the epidermal layers, on the outside layers. So what happens to the outside layers of the plant? They get broken. Avi virigipotayi annamata. It means the distance between the cells, they will increase. Are you able to understand? That's what he is trying to say. So because the activity of vascular cambium, outer cortical and epidermal layers get broken. They will break down. So it is to be replaced to provide new protective cell layers. So new layers should be developed, no? Hence another meristematic tissue called cork cambium develops, which is called phloem, usually in the cortex. Are you able to understand ma? So therefore another cambium. And what is the name of that cambium? Cambium means what ma? Layer of cells, that's all. Cambium means, you should not be confused. Cambium means a layer of cells. So another layer called cork cambium develops in the cortex. And where is this cortex? Inner layer to epidermis is called cortex. So in the cortex, another layer called cork cambium develops to replace the broken cells. Are you able to understand ma? So that is the function of cork cambium. So see ma, here this is the cork, no? and inside the cork, what you find inside the cork, you will find cork cambium. That is the next layer of cork is called as cork cambium. Okay, that's it. Understood, Nama? Next, phellogen. What is meant by phellogen? This cork cambium is nothing but phellogen. So phellogen is a couple of layers thick. Couple of layers thick means what, ma? Two layers are there. So in cork cambium you will find two layers. Two layers are present. It is made up of narrow, thin-walled and nearly rectangular cells. So what is the shape of the cells? Narrow shells. Walls are very thin and nearly rectangular cells. This type of cells. Clear, Nama? So phellogen cuts off cells on both sides. So outer cells differentiate into cork cambium, phloem, and inner cells differentiate into secondary cortex, which is called phelloderm. Okay, ba? So cells of secondary cortex are parenchymatous. So this is the condition. So have you understood what is happening here? Phellogen. Phellogen means what? Cork cambium. Already we studied about that one. No? A new layer of cambium develops to replace the broken cells. Clear, Nama? So this phellogen, it again divides into inside also, it divides on the outside also. So the division which is taking place on the inside, they will form what? They will form secondary cortex. The outer cells differentiate into cork cambium or cork, not cork cambium, cork. Outer cells of cork cambium, they will form cork. And inner layers of cork cambium, they will form secondary cortex. Cork is called phloem. Secondary cortex is called as phelloderm. Are you able to understand, ma? And secondary cortex are made up of which kind of cells? Parenchymatous cells. Are you able to understand? See, for example, if this is the cork cambium, what is this one? This one, let us call it as cork cambium. Okay. So it divides on the outside, and on the outside, which color shall I use? I will use this color. On the outside, you will another layer is formed. This layer is called what? This layer is called cork. And on the inside also, you will find another layer. On the inside also, you will find another layer. And what you call this layer? This layer is called as secondary cortex. Are you able to understand? Cork is called what? Cork is called as phloem. Okay, ma? And secondary cortex is called as phelloderm. So this terminology you should remember. Are you able to understand? Cork cambium is dividing in the inside also, it is dividing on the outside also. Clear? Outside is called phloem, inside is called as phelloderm. Phloem is also called as cork, phelloderm is also called as secondary cortex. And secondary cortex is made up of which kind of cells? Parenchymatous cells. Clear? That's it. Okay, ma? Next. So the cork is impervious to water due to suberin deposited in the cell wall. So the cork which is formed because of cork cambium, it is impervious to water. Impervious to water means what? It doesn't allow water because of a material deposited which is called as suberin. Are you able to understand, ma? Arthamautundi kada? Cork. Cork is developed from where? From the outer divisions of cork cambium. So this cork, it is impervious to water because of which material, ma? Suberin. So phellogen, phloem and phelloderm are collectively called as periderm. Okay, that is phellogen, cork cambium. Phloem means cork. Phelloderm means secondary cortex. All of them, they combinedly they are called as periderm. Okay. So due to activity of cork cambium, pressure builds up on the remaining layers peripheral to phellogen, and ultimately these layers die and slough off. So here also, because of cork cambium also, pressure is developed on the outermost layers. Which are the outermost layers? Cork, epidermis, all these are outermost layers. No? So on these outermost layers, again pressure will be developed, again they will break down and they will fall to the ground. Slough off means what? They will fall to the ground. Okay, ma? So here one point which you should understand is pressure always develops on the outside. Ma? Pressure never develops in the inside. Because of cambium, because of vascular cambium also, pressure was developing on the outside. And because of cork cambium also, pressure was developing on the outside. Fine, that's it. Okay, so bark is a non-technical term that refers to all tissues. Okay, so periderm and secondary phloem combinedly you can call them as bark. What is meant by periderm already we have studied now. Combination of phelogen, phloem and phloem is called periderm. And periderm plus secondary phloem you can call it as bark. Okay, ma? So exterior to the vascular cambium. It means they are beyond the vascular cambium, nama. From the vascular cambium, you can find the secondary phloem. Then you can find the secondary cortex. Then you can find the cork cambium. Then you can find the cork. Are you able to understand? So all this is called as bark. Okay. So therefore, bark is a combination of what? Bark is a combination of secondary phloem plus secondary cortex, plus cork cambium plus cork. So this totally you can call it as the bark. So bits will be asked like this one. Clear? So or just you can say bark is a combination of periderm plus secondary phloem. Okay, and periderm means what? Periderm means secondary cortex, cork cambium, and cork. All of them it is called as periderm. Understood, Amma? That's it. So then this cork cambium again, bark. It is of two types: early bark and late bark. Early bark means it is formed early in the season. Late bark means what? It is formed towards the end of the season. So bark also you can divide into two types. Clear, Nama? Early bark, late bark. Early bark is soft, late bark is hard. That's you have to by heart. Clear, Nama? Okay, we will stop up to here, ma. We will continue in the next class. So next year start cheyali manamu lenticels. మీకు ఎంత ఉందిరా ఇది? అంతేనా? ఈ మాత్రం దానికి సెకండ్ మళ్ళీ ఇంకో క్లాస్ ఉంది. Only 2 slides are there. We will complete it. Okay, ma. See. So next let us see what is meant by lenticels. So at certain regions, phelogen cuts off closely arranged parenchymatous cells on the outside. These cells rupture epidermis, forming a lens-shaped opening, which is called as lenticels. Are you able to understand, ma? So what is meant by lenticels here? Here you are seeing one opening, no, ma? This opening you are seeing here, no? This opening is nothing but a lenticel. Are you able to understand? And what is happening here? Phelogen. What is meant by phelogen? The cork cambium is called phelogen, no? Clear, no? So this cork cambium, it cuts off closely arranged parenchymatous cells on the outside. Why? Because cork cambium, it develops pressure on the outside layers, no? So because of this pressure, some of the cells, they will get separated and a gap will be created. Are you able to understand, ma? These cells rupture epidermis. So because of this pressure, they will destroy the epidermis. What is meant by epidermis? Outermost layer is called as epidermis, forming a lens-shaped opening which is called as lenticels. Clear? And where they occur? They occur in most woody trees. That is in very, very big trees where you find lot of wood, you can find this lenticels, openings in the stem. Okay? So lenticels permit gaseous exchange between the outer atmosphere and the internal tissue of the stem. So what is the function of lenticels, ma? Gaseous exchange is the function. That's it. Clear? So secondary growth in roots. In roots also we will study the secondary growth, ma. Okay. In a dicot root, vascular cambium is completely secondary in origin. Are you able to understand? So in dicot root, when you will find secondary cambium, ma? Secondary cambium is secondary in origin. Secondary in origin means what? In primary root, you don't find the vascular cambium. Are you able to understand? In dicot root, in the primary growth, you will not find the vascular cambium. You will find vascular cambium in the root of dicot plants only in the secondary growth. Okay? So this is another important point. So it originates from the tissue located just below the phloem bundles, a portion of pericycle, above the protoxylem, forming a complete and continuous wavy ring. It later becomes circular. Okay, ma? Further events are similar to those of a dicotyledonous stem. Are you able to understand, ma? So therefore, it is developed from where, ma? It originates from the tissues located just below the phloem bundles. So group of phloem will be there, nama. From this phloem bundle tissues only, this vascular cambium will develop, which is a part of a secondary growth. Okay, ma? And it is above the protoxylem, forming a complete and continuous wavy ring. So it is in the formed in the form of a wave, wavy ring like this. Clear? Later it develops into a circular ring. Are you able to understand? So the first vascular cambium formed in dicot roots. It is of its shape. It is in the shape of waves. Then finally it develops into a ring. Clear, Nama? So then further events they are similar just to that of a dicotyledonous stem. How dicotyledonous stem will behave? So the roots also they will behave in the same manner. So secondary growth also occurs in stems and roots of gymnosperms. Secondary growth doesn't occur in monocotyledons. Clear, Nama? So secondary growth is possible only in dicots only, no? Because in dicots only you will find a cambium. In monocots you don't find any cambium. So secondary growth is not possible in monocots. Secondary growth also occurs in stems and roots of gymnosperms. Okay, ma? So that's about the secondary growth, and that's about the topic called anatomy of flowering plants. Understood, Nama? Very, very easy topic. Just you have to revise it and you have to connect it. ఒక పాయింట్ ఇంకో పాయింట్ కనెక్ట్ చేసుకుంటూ చదవాలి. Then it becomes very easy. Okay, ma? Thank you. We'll see in the next class. సో రేపు మీరు సుప్రజ, ప్రణీత్ మొబైల్ ఫోన్స్ తెచ్చుకోండి. ఓకేనా? ఈ చాప్టర్ చదువుతాం రేపు. సుళ్ళలంకటం వాళ్ళు వద్దు, మీరు మాత్రమే తెచ్చుకోండి. సూడో అనే పదానికి కొంచెం అర్థం ఉండాలి కదరా.